Each and every one of you, I hope each and every one of you are doing well, safe and sound and doing extremely well in life. So good evening each and every one of you. Welcome to Vedantu NEET English. This is Basavaraj. I'm your biology master teacher. So students, I hope before we start anything, I hope you're able to hear my voice and you're able to see the presentation behind me. So students, if you're able to hear my voice and see the presentation behind me, a quick thumbs up in the chat or a fire in the chat, or everyone today, green hearts in the chat, green hearts if you're able to see the presentation and hear my voice. So good afternoon each and every one of you. Let me know how is everyone doing in the chat. Good evening, good evening. No, I'm not stuck in any traffic. I'm in here in the studio live with all of you. So before I start the video, I want each and every one of you to like the video, share the video and subscribe to the channel, that is Vedantu NEET English. So students, before everyone joins, let me ask you a simple question. So all of you know, today we are doing plant growth and development. Yes, but I ask you a simple question to all of you. Is growth a defining feature of living organisms? A simple question, right? Is growth a defining feature of living organisms? While I wait for everyone, let's see how many of you can answer this simple question. Hello Gautam, hello Teju, hello Shalini, hello Manisha, hello Rohit, each and every one of you. Good evening, all one and all. So students, today we'll be doing the chapter Plant Growth and Development. This is the final chapter of the plant physiology. I hope all of you have learned photosynthesis, respiration. Today is the last chapter. So yes, all of you are saying, no sir, completely wrong. Yes, growth is not a defining feature, but it is just a character. Answer to that question, I will tell you in the next 2 minutes. So students, can I start? Can I start the class today? Each and every one of you, thumbs up in the chat, green hearts in the chat. If I can start today's amazing class, the last chapter of your plant physiology. Can we start? Yes, it is not a defining feature. Exactly. Easiest chapter. Yes, it is easiest chapter. But students, how many questions you can expect from this class? Right? From this particular chapter, how many questions? So students, initially, we used to get at least 2 to 3 questions. Yes, before the syllabus reduction, we used to get 2 to 3 questions. But after the deletion of the first 2 chapters, the plant physiology game has changed. Yes, the game has changed completely. Now you can expect at least 3 to 4 questions. 3 to 4 questions from this chapter alone. 3 to 4 questions from this chapter alone. So students, let me know in the chat how is going to be today's class. And before that, everyone, I see all of you becoming confused, as Gopika ma'am says, in liking the video. So like the video and we will start today's class, that is plant growth and development. Let's not waste any time. So here we go. Students, we'll be starting with growth. So before I start today's class, I want each and every one of you to please have a notebook next to you. Yes, Gautam, hello Sharat, please keep a notebook next to you and please write down each and every point I'll tell you. Why? Because students, I'm taking direct points, direct notes from your NCERT. I'll be decoding the NCERT today. After my class, just go read the NCERT once and you will know exactly what you need to know for your examination. So can we start? So all of us know we'll be starting with growth. Then I'll be teaching you the different parts of growth. That is, how do we measure growth? What is the meaning of growth? What are the different phases of growth? Every single point here. Then I'll be teaching you differentiation, dedifferentiation and redifferentiation. Yes, the proper difference I'll be telling you. Then we'll be focusing on the development. Yes, growth and development, right? So you'll be focusing on development. Then at the last part, the most important part of this chapter is the hormones. In total, we have 5 hormones and we'll be studying all the 5 hormones in today's class. Can we start? Can we start, students? Gautam is asking, my bio syllabus is completed. Gautam, remember that. You might have finished the chapter. You might have finished the entire bio. But remember, the more you revise, the more the concepts become clear in biology, the more marks you'll get. Can everyone agree with that? Can everyone agree with me on that? The more you revise, the more questions you solve, the more confident you become, the more well versed in this particular chapter you become. So with that being said, let's start with the first part of this chapter, that is, what is growth? What is the meaning of growth? Now everyone knows, all of you know that growth is not a defining feature of your living organisms. I'll quickly explain you that. Why? Students, we know this particular growth, we know growth can be seen in the case of both living organisms as well as non-living organisms. So what are you saying? So can anyone in the chat give me an example for growth in your non-living organisms? Let me tell you. In your living organisms, yes, we normally see growth. In living organisms, we normally see the growth. Yes, I have grown in my height. My cells number has increased. The cell division has happened. But students, in your non-living organisms, how do you see non-living things? How do you see the growth? Let me tell you a simple answer. Listen to me very carefully. This is your mountains. Yes, sir, this is the mountains. This is the current height of a mountain. Yes. During winter season, can I see snow on this in your Ladakh regions? Yes, sir. In the Leh Ladakh as well as in the much more Arctic regions, can I see a accumulation of snow on top of the mountain? Yes, sir, we can see. So can I tell, did the mountain actually grow? Yes. So this type of growth which happens in the non-living things is called as extrinsic growth. Extrinsic growth. But in the case of humans, we see, or any living organisms, we see intrinsic growth. That is the difference between living as well as non-living, which both of them show the growth. Now I'll tell you definition. Everyone write down the definition with me. Yes. When the board series will launch very soon, students, do not worry. We are with you. We are with you till the end of your NEET examination. We have board series coming up. In fact, we have a new surprise for all of you. New surprise for all of you, which will come out this weekend. Okay? Stay tuned for that. Now, let me write the definition for all of you. Let me write the definition which all of you should also write down for growth. Now, can I call growth? Everyone in the chat, can I tell growth? It is an irreversible change. It is an irreversible change. Yes, sir. It is an irreversible change. Now, it is a change which is happening. You cannot reverse it. Now, I'll give you an example. Let's take someone from the class. Harsha. Now, Harsha was in 8th class. When Harsha was in 8th class, he was 5 feet tall. Now, Harsha, when he is reaching class 12th, he is around 6 feet tall. From 5 feet to 6 feet tall. Now, can anyone in the chat tell me, is that growth? Is that a form of growth? The answer is yes, sir. That is a growth. It is an irreversible change. It is a permanent change. Yes, sir. Harsha grew tall. Now we'll take one more example. Take me an example. 10 years back, I was around 50 kgs. 10 years back, I was 50 kgs. Now I am 65 kgs. Can anyone in the chat tell me, is that also a form of growth? Everyone, can anyone in the chat tell me, is that also growth? I was 50 kgs 5 years back. Currently, I am 65 kgs. Is that growth? First, tell me in the chat. Is that also growth? Now, when I say permanent, irreversible, it's a permanent, irreversible change. Now, what is this change we are talking about? This change, in the first example I told you, that is change in the size. Yes, sir, there can be a change in the size. There can be change in volume. Yes, sir, there can be change in volume. There can be change in mass, or they can also be change in the case of length. Yes, sir, there can be any form of change. And whenever this change is happening, it has to be irreversible. Now I ask a question. The weight, I was 50 kgs before. Now currently I am 65 kgs. Now suddenly I decide, no, I will not eat any type of food. I will go to gym. I will only diet and I'll bring back my weight. I will bring back my weight to 50 kgs. Bring back my weight to 50 kgs. But sir, you told me growth has to be irreversible. But I just reduced my weight. So I'll ask you the question again. Can I call growth, that is increase in weight, as a growth? The answer should be no. The answer should be no, not yes. Because that is reversible. A weight of a person can be reversible, and hence the answer has to be no. No, sir, that is not a growth. Example, height. Yes, sir, I'm growing tall, but is it reversible? No, that is growth. Your NCERT gives another example. NCERT gives an example as, you take a container. In the container, you add a stick, and this is water. The stick will absorb the water. Yes, sir, the stick will absorb the water and the stick will swell. Is that a growth? The answer should be no. Why? Because after some time, the stick will shrink again. The water will evaporate. That is reversible. So that is not a growth. That is not a growth. One more example which is given in your NCERT is the cells, your stomatal cells. The stomatal cells will absorb the water and they become turgid. Is that a growth? The answer should be no. That is also not growth, because as soon as your stomatal cells lose the water, they become flaccid again. Yes, sir, that is not a growth. So students, please write down, please remember this. A growth should be always and always irreversible, and it has to be permanent. It has to be permanent. Clear? Is everyone clear on that point? Just say clear and I'll teach you the next concept. Here we go. Is that point clear? So I'll write down few points about growth here. I told you growth is irreversible. Yes, sir, growth is irreversible. Can I call growth as more of anabolic process? Anabolic or catabolic? Why, sir? Why is it more anabolic? Because you are forming something. As we grow, we are forming. We are from simple to complex substances. So can I call it anabolic process? Yes, sir, you can call it anabolic process. When you're growing, when an organism is growing, do you require energy? Yes, sir, do you require energy? Tell me. You eat food. The food gets broken down. Energy is obtained. So can I tell, in the growth, we require energy? Yes, sir. Energy is required for growth. Yes, sir. Energy is obviously required for growth. The last point here. The last point about growth is, can we measure the growth? Can we measure the growth? The answer should be yes, sir. I was 5 feet. Now I am 6 feet. That is growth. You're quantifying it. Yes, sir. The last point I want all of you to write down is, growth can be measured. Growth can be measured means it can be quantified. The growth can be quantified. That is the last point about growth. Is everyone clear now? All of us know the basic difference. What is the meaning of growth? Yes, sir. All of us know what is growth now. Growth is an irreversible change. It is a permanent change. It is more of an anabolic process than catabolic process. Growth requires energy. And lastly, a growth can be measured. A growth can be measured. So with that, we will start the chapter that is growth and development. Right? Plant growth and development. So students, since we are talking about your plants, let's talk about germination. Now, I told you what is growth, but then what is the meaning of germination? Germination is nothing but growth of a seed. Yes, sir. We are talking about plants here. So obviously we need to talk about germination. What is germination? Growth of a seed. Growing of a seed here. We have a seed here. The seed is growing of a seed. If you want to write it very specifically, it's a growth of the embryo. It is growth of the embryo which is inside the seed. As soon as the seed grows, it develops a radical. It develops a radical towards the down, and towards the air, it develops a plumule. Radical and plumule, and the plumule gives rise to your shoot, and the radical gives rise to your shoot. That is your growth in your plants. I hope that point is clear to all of you. If you look at your NCERT right now, this is a diagram given here. See, this is your small cute seedling. That particular small cute seedling is growing. Yes, sir, it is growing. What is growth? It is an irreversible change. Yes, it is growing into your radical and plumule. This is your seed coat. Then we have your epicotyle and hypocotyle. Then we have a beautiful shoot and root system. Students, I'll be following your NCERT paragraph by paragraph. So keep your NCERT next to you. Keep your register notes in front of you and keep the video before you and write down all this. Okay, please keep on writing all of this. Now I'll be telling you about the next concept is, which is given in your NCERT is, in plant growth. We learned about growth. Now we'll be completely focusing on your plant growth. Complete focus on your plant growth. Please write down with me. First thing I can tell you about plant growth is completely unlimited, also known as indeterminate. Plant growth is completely unlimited. You might have noticed your height, my height, everyone's height stops growing at 22 years maximum. Yes, sir. After 22 years, no one grows. Let me know in the chat if some of you can grow after 22 also. Okay. But 90%, 99% of the population on earth will completely stop growing at 22 or 21. But what in the case of plants? Plants, you might have noticed, they will actually keep on growing throughout its lifespan. Throughout the lifespan, a plant will keep on growing. It might not be just your vertical growth. Plants can keep on growing even horizontally. So can anyone, any smart kid in the chat can tell me what is so different about us and the plants? How are we different from the plants? How are plants able to continuously grow, and how can we not grow at all? What makes plants so special? The answer is very simple. It is meristematic tissue. What it is? It is called as meristematic tissue. All of you write down. Because plants have something called as meristematic tissue. Meristematic tissue. Now what is this meristematic tissue? Students, if you know the anatomy of flowering plants, we know meristematic cells or meristematic tissue is a continuously dividing cells. Yes, and we also know we have two types of meristematic cells. Yes, what are the two types? The complete meristematic tissue can be divided into two parts. First one is called as your primary meristem. It's called as what? Primary meristem. And the second one is called as your secondary meristem. Secondary meristem. Yes. Now what is the basic difference? All of you should know the basic difference here is the primary meristem. The primary meristem is found at the origin, at the beginning of the plant development. As soon as the plant was developing, the primary meristem was there. So can I write the primary meristem is from the origin, from the beginning, all the way from the beginning it is present. But what about secondary meristem? Secondary meristem is later formed. Later formed. Yes, it is later formed. Now we also know your primary meristem. Your primary meristem is responsible for the increase in length of a plant. Yes, sir. Primary meristem is involved in the increase of the length of the plant. Yes. Length can be increased. Increase in length. It is responsible for increase in length. Yes, sir. Now, what about secondary meristem? Secondary meristem is responsible for your lateral growth. It is responsible for the lateral growth. Lateral growth, also called as the width, increase in the width or increase in the girth. Increase in the girth. 1 second, students. Increase in the girth of the plant. Now I'll tell you what exactly it is. Increase in the girth. Now I'll tell you the basic difference. Last difference here is in the case of your primary meristem. Primary meristem can be seen in both as in monocots as well as dicots, both. But in the case of your, listen very carefully, in the case of your secondary meristem is only and only found in your dicots. Write down. Your primary meristem can be found in both, that is monocot and dicot. But in the case of your secondary meristem, it is only found in the case of your dicots. Only in the case of dicots. Now, if you look at the diagram here, your NCERT diagram, see this. If you look at the NCERT diagram, can you see this shoot apical meristem as well as root apical meristem? Root apical meristem. This is nothing but your primary meristem. It is also part of your primary meristem, which is responsible for the increase in length. This is nothing but your SAM, shoot apical meristem, which increases the length of the shoot. Yes, sir, it increases the length of the shoot. But we also have that is called as root apical meristem. Can I call it also RAM? RAM, not RAM, RAM. The RAM is responsible for the increase in the root length. Yes, increase in the root growth. Now, what about secondary meristem, sir? Where is the lateral meristem? The secondary meristem here is here. Can you see? The vascular cambium. That is part of your secondary meristem, which is responsible for the width. This is how the plant will grow. The thickening. Also responsible for the secondary growth. Same vascular cambium. Responsible for the secondary growth. You can see increase in width. Clear? The first point here is in the plant growth, that is a plant shows unlimited or indeterminate growth. Indeterminate means ultimate growth, means the plant keeps on growing. How? Because of the meristematic tissue. We have two types called as primary meristem and secondary meristem. Primary meristem is present from the origin. As soon as the plant is born, it has the primary meristem. But the secondary meristem is later found. Yes, sir. This primary meristem is responsible for the increase in length of the shoot as well as the root. While in the case of your secondary meristem, lateral growth, increase in the girth. Then primary meristem is found only in the case of your monocots, while your monocots and dicots, while your secondary meristem is only and only found in the case of your dicots. Example again, example for your primary meristem is your SAM and RAM. Examples for your secondary meristem is your, where is it? Vascular cambium. That is the first point which is given in your NCERT, that is the plant growth is ultimate or indefinite, unlimited. Now the second point. All of you understood the first point? Let me know in the chat if you understood the first point. That is the unlimited growth. Now second point. The second point here is the plant growth is open. Plant growth is open. Now what is the meaning of open? Open and close. The meaning here is very simple, students. Listen to me very carefully. The meaning of open here is a plant will keep on adding new cells. A plant will keep on adding new cells. New cells are always added. What is the meaning of open? New cells are always added. Please write down this. Always added. New cells are always added. But in the case of humans, it is closed. New cells are not always added. They are rarely added. Whenever there is requirement, it is added. But in the case of plants, they are open, means new cells are added all the time. That is the second point. Now, what is the third point which I want all of you to understand is, in the case of plant, the plant growth is, this is very important. Plant growth is localized. Now, what is the meaning of plant growth is localized? Listen to me very carefully. Imagine, all of you imagine now. I want everyone to imagine this. A plant is growing. You might have seen, I just told you, plant will only grow wherever meristem is present. That's why a plant will always grow from the top. That is, a shoot apical meristem is present, a plant is growing. Whenever there is root apical meristem, the plant will keep on growing bottom. Yes, lateral meristem, it grows. But in the case of humans, the growth is completely uniform. That is why we never see a small baby with only arms are growing. You never see that. Or only head is growing in the human. No. Only the legs are growing. No. In the case of humans, it is uniform. But in the case of plants, the complete plant growth is localized. Plant growth is localized, means at very specific spots, very specific local areas, the plant is growing. The example is your SAM and RAM, that is SAM and RAM. Clear? That is the third point I want all of you to understand. Now, next point I want you to understand here is, this is meristem. Wherever meristem is present, in those places you can see the growth. The next point I want all of you to understand, the last point in your plant growth is the plant, right? In case of your plants, the organs, the plant organs are always added. What is the meaning, sir? How can you say that? Plant leaf. All of you know the leaf, right? Leaf is an organ. Yes, flower is an organ. Do you see new leaves every season? Do you see flower every season? Yes, sir. I see all the time flowers. What is the meaning of that? The meaning of that is, in the case of plants, the number of organs keep on increasing. The number of organs keep on increasing. Example is your flower. Remember, flower is a reproductive organ. Along with that, your photosynthetic organ, that is your leaves. Those are the 4 points about your plant growth. The first point was what? The first point was your unlimited or indeterminate growth. Students, remember, in your examination, they will directly ask you, what is the meaning of indeterminate? You should know the meaning of indeterminate is unlimited growth. First point. The second point we need to understand here is, plant growth is open. What is the meaning of open? It means that new cells are always added. Yes, sir, we know plant cells are always added. The third point is, plant growth is localized. Only in very specific areas. Wherever the meristem is present, in those areas only you will see plant growth. Clear? The last point here I want all of you to note down is, plant, the number of organs keep on increasing. The organs such as leaf, the leaves are added always. The flower, the reproductive unit is always added. With that, that is the end of your plant growth. 4 points. I hope all of you know it. No need to by heart. Understand it. Okay? Now, the next point I want all of you to understand is, I told you, growth is measurable. Yes, sir, I agree with all of you. You're telling me, yes, sir, we can measure the growth. But students, what I'm trying to tell you here is, whenever a cell is growing, whenever a cell is dividing, the protoplasm is increasing. Now, what is protoplasm? Do all of you know what is protoplasm, or should I tell you? Do all of you know what is protoplasm? Protoplasm is the living part of a cell. Yes, sir. Protoplasm is the living part of a cell. What is the meaning? Inside your plasma membrane, inside the inside the plasma membrane, whatever is there, that is your protoplasm. That could include your cytoplasm, cell organelles, and nucleus. All of them together is your protoplasm. Clear? Now students, I told you growth is measurable. Now if this is your cell, if this is the cell, this is your protoplasm inside. Yes sir, this is the protoplasm. Now I told you, you agree with me that a cell is increasing in size. Now what is the meaning of that? The meaning of that is the protoplasm is increasing. Yes sir, the protoplasm is increasing. So can I draw like this? Bigger cell? Yes sir, a bigger cell. Can I see the protoplasm? Yes sir, the protoplasm has increased. So what do I call it? I call this protoplasmic growth. I call this protoplasmic growth. But students, remember this very carefully. Listen to me words. Listen to my words very, very carefully. This particular protoplasmic growth is extremely difficult. Very, very difficult to measure. Very difficult to measure. That's why we have certain alternative methods. Okay, that's why we have certain alternative methods, because this particular protoplasmic growth is very, very difficult to measure. That's why we have different measurements. That's why we have what? Different growth measurements. The first one, which is given in your NCERT, is increase in, what do you measure? For write point wise. Sure, write point wise. It will be easier for all of you to write down also. The first one which is given in your NCERT is increase in the number of cells or increase in the mass. Yes, so increase in the size and increase in the number. The first part about your growth is measurable. The first point is increase in the size as well as the increase in the number. Write down the first point, all of you. The first one is what? Increase in cell size and cell number. Yes, increase in cell size and cell number. Okay, I don't think that is visible. I'll write it one more time. Handwriting is bad on this board. Usually my handwriting is good, but and cell number. Now do I have an example for this? Your NCERT clearly tell one example. NCERT says one example for each one of them. For example, for increase in cell size, NCERT is giving an example of your watermelon. Initially, listen to me very carefully. Initially, this watermelon was, this is your watermelon. Initially, this is your watermelon. Now, this watermelon, each cell will grow so much. That is, each cell is growing approximately 3.5 lakh times. 3.5 lakh times. The cell is growing. That is an example of your growth in the cell which we can measure. That is increase in cell size. That is the example which is given in your NCERT is the watermelon times. Now what about your increase in cell number? Your NCERT gives another example. That is your maize. Maize. All of you know maize. All of you know what is maize. Let me know, child, what is the name of the maize? What do you call maize in your language? In Kannada, I forgot. But let me know, child, what do you call maize in your language? Now in this case of your maize, the ram, that is root apical meristem. In the case of your maize, the root apical meristem will increase in number. It will increase in number almost 17,005 times per hour. Almost 17,500 per hour. Times will increase. That is the increase in cell number. So the first point here, the growth is measurable here is increase in the cell size, the second and increase in the cell number. Your NCERT is telling you two examples here. The first example is increase in cell size is your watermelon, which is increasing approximately 3.5 lakh times its original size. In the case of your maize, the maize ram, the root apical meristem is increasing at least 17,500 times per hour. That many number is increasing. So that is the quantifiable number for your cell size as well as your cell number. So that is that. The second point I want all of you to understand here is we can quantify, we can measure increase in fresh weight, we can measure increase in fresh weight and dry weight The next one is increase in length. Yes sir, we can measure increase in length. The example increase in length, the example which is given in your NCERT is the pollen tube growth. The pollen tube growth, that is increase in length. The next point I want all of you to focus here is the fourth point, that is increase in surface area. Increase in surface area. Initially we had a small leaf. And the small leaf has grown this big now. Can I quantify it? Can I measure the increase in surface area of a leaf? Yes sir, we can measure the increase in surface area of a leaf. So these are the 4 points for your growth is measurable. The last point I want all of you to know is increase in volume. Increase in volume. So the answer to this first question here, growth is measurable. Yes sir, growth is measurable. But what is the actual growth? The actual growth is the protoplasmic growth, the increase in the protoplasm. But is it easy to quantify this? No sir, it is very difficult to measure this. But do we have an alternative to this? Yes sir, we have alternative to this. Example is increase in cell size. Example is watermelon. Initially from the small cell it is increasing approximately 3.5 lakh times increase in cell size. What about increase in cell number? In the case of your maize, around 17,500 per hour it is increasing. The cell number is increasing in your RAM. The next one is increase in the fresh weight and dry weight. Yes, fresh weight of a leaf and a dry weight of a leaf. Then one is your increase in length. What is increasing? Example is pollen tube. If you add the sucrose, the pollen tube is growing. Yes, that is increase in length. Then we have increase in surface area. In the case of leaf, you have a small leaf. The small leaf is growing. It is becoming a large leaf. Increase in surface area. The last point here is increase in volume. Yes, the fruit is increasing in volume. Any single part of your plant body is increasing in volume. So these are the 5 points, that is growth is measurable. The answer is yes, the growth is measurable with different parameters. Clear? Any doubt till now? Yes, 10% sugar solution. Any doubt till now? Watermelon is increasing in size. Watermelon is small watermelon. It is so cute initially. It is becoming that big, right? Any doubt till now? You can ask me, students. Any doubt till now? Growth is measurable. Done. I will teach you next topic now. Okay. Can we start the next topic? The next topic which I want all of you to do is phases of cell. Oh, we have a question here. We have a question. All of you. Let's see the question. Let's see the question, all of you. Students, if you are actually understanding today, if you are following what we just did from past 45 minutes, I want everyone to drop in a green heart. All of you, every single student who is watching there should drop a green heart and solve this question. What is the question? The growth of the following statement, which of the following statement is or are true with respect to the plant growth. With respect to the plant growth, the root and the stem of the plants are descendants of a single cell. The roots and the stem, the root and the stem are coming from a single cell. Second one is leaves and fruit show indeterminate growth. I deliberately didn't tell you a point here, which I will explain now. Okay. The next one is plant development includes both growth and differentiation. Let's see who can answer this question. I'll give you 10 seconds. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Time's up. The answer is actually D, 1 and 3. Because we know the root as well as stem of the plant descend from a single cell. Yes, we have a single zygote. Zygote develops into embryo. Embryo differentiates later on and forms the root and the stem. Yes, it forms the root and the stem. This is correct. The leaves and the fruit show indeterminate growth. They do not show indeterminate growth. It does not show indeterminate growth. What is the meaning, sir? We know the leaf will not keep on growing. Only the shoot and root meristem. Wherever we find RAM and SAM, we find the indefinite growth. But in the case of your leaf and fruit, you do not see indefinite growth. Clear? In the case of plant development, it includes both growth and differentiation. A plant is growing and it is differentiating later on. Does anyone has a doubt why this is not option number C? Because we know root and stem develop from a single cell. That is your zygote. Clear? Any doubt here? Can we start with the phase of growth? Can we start with the phase of growth of a plant? So we know this is one cell. Now this cell will divide. Yes, sir. This is a normal cell division. Yes, sir. The cell is dividing. The first step in the cell division is actually division. Yes, it is division. So the name which is given for the first phase in your cell division. The first phase in your cell division is nothing but your meristematic phase. Meristematic phase, where the cell shows active division. The second phase of a cell division is second phase of your growth is the elongation of a cell. Yes, the second phase is your elongation. Second phase is your elongation. Second phase is your elongation phase. Yes, it's elongation. That is your cell elongation. And the final phase which I want all of you to understand is the cells are dividing, they're elongating, and what happens then? Think of you as an example. As you were born, you were growing rapidly in the starting. Yes, you were growing rapidly in the starting. That is, you were dividing, then you were elongating, and finally you become wise. You get all the wisdom in the world. That is maturation. The third phase in your growth is your maturation. Clear? Can we write down the properties here now? Can we write down a few properties of your meristematic phase, elongation phase, and maturation phase? I will write it for you. Please note down, and I'll teach you also what it is. Do not worry, the entire PDF of today's session will be given to you on the Telegram channel. Can we start? RAM means root apical meristem. Yes, root apical meristem. SAM means shoot apical meristem. Now, let's start with the meristematic phase. I will write down the few properties of the cell. Please write down with me. Okay, all of you write down with me. Okay. Now, can I call this phase, the first phase, as, write it as A. Is this the first phase? So first point is, it is the first phase. Simple. It is the first phase. Now it's called as meristematic phase. So can I find here meristematic tissues? Yes sir. The second point is going to be it has meristematic region. It has meristematic region. It has meristematic regions. What are the meristematic regions here? Someone in the chat was telling me it has to be your SAM and RAM. Yes. Now next one here is, what about the cells? How are the cells? Are they large cells or they tiny cells? The answer here is, since it is a starting stage, the cells are just dividing. So cells are going to not be large. The cells in the starting stage are going to be tiny and cute, small cells. So they are very small cells. Very small cells, they have to rapidly divide. Remember in your cell cycle and cell division, whenever the cell division is happening, we are forming so many cell organelles and we need a space for everything. So what about the protoplast? In the case of dividing cells, there is always, remember this, write down this, dense protoplast. Remember this, in the case of your dividing cells, always it is dense protoplast. Very dense protoplast. Now what about vacuoles? Sir, they are just dividing. They just start dividing. Do they require a storeroom? Imagine you just started, your NEET journey. Okay, do you need around 10 different notebooks to write? No. You just need one notebook, or you just need few pages. Just starting something, you're writing a book. Imagine you're writing a book. Do you need 10 extra copies of book? No. If you're just starting the division, do you need large vacuoles? Do you need many vacuoles when you're dividing? There's no food storage only. Why do you need storage room? So can I tell, in the case of your meristematic phase, the cells will have almost no or vacuoles absent? Yes, vacuoles are almost absent here, or very less vacuoles. Vacuoles are almost less. What about the nucleus? The nucleus is conspicuous, large nucleus, very large nucleus. Large nucleus can be seen here. Now what about cell wall? What about the cell wall? Since it is a just starting stage, do you think we have primary cell wall or secondary cell wall? Everyone, you should only tell me, what is the meaning of dense? Dense means compact. Dense means very compact and very dense. Everyone should tell me in the chat, since it is the starting phase, what will be a cell wall? Do we have primary cell wall or do we have secondary advanced cell wall now only? No sir, it is a starting stage. So we have just a primary rudimentary cell wall. So can I tell primary cell wall? Primary cell wall. Yes sir, a very primary cell wall. Now the cells are just dividing. The cells are just dividing. They need to talk to each other. They need to be in contact with each other. So last point here I want all of you to write down, which is also given in your NCERT, is your multiple plasmodesmatal connections. Plasmodesmatal connections. So students, in the case of your meristematic region, in the case of your meristematic phase or meristem phase, you have first phase. Yes, all of you know. We have meristematic regions, that is your SAM and RAM. In the case of your meristematic phase, of your growth phase, we have small cells with dense cytoplasm, very compact cytoplasm, or your protoplasm. Then we have vascules are completely absent. We do not need storage here. Then we have large nucleus, very conspicuous nucleus. Then we have a primary cell wall. Initial phase, a very primary cell wall. Then we have multiple plasmodesmatal connections. Those are the connections between the cells, a cell to cell channels. Plasmodesmatal is nothing but channels between the two cells. So these are actually all the points which you need to write down and remember for your meristematic phase. Clear? Clear? Can we go to the next point here? Can we go to the next one? That is your elongation phase. The next one is your elongation phase. Now the cell has started to divide. Now it is elongating. Means what? It is increase in size. Yes, increase in length. So the first point I want all of you to write here is, it is the second phase. It is the second phase. Here the cell is divided. Now the cell will increase in length, increase in size. Yes sir, in the case of your second phase, there is cell length increase. The length of the cell is increasing, and also the size. The length is increasing, the size of the cell is also increasing. Size is also increasing. Now what about your vacuoles? Now in this case, vacuoles will start to appear. Vacuoles will start to appear. It's called as vacuolization. The vacuoles will start to occur. Vacuoles will occur. Means their vacuoles are just starting. Because we are increasing in cell number, we are increasing cell size. So we need more vacuoles now. We need something for storage. So vacuole number is increasing. The last point here is, not last but one point, the secondary cell wall will appear now. The secondary cell wall will start to appear. That is, over the one cell wall, we have the secondary cell wall will start to appear now. The last point here is, the last point I want all of you to understand here is, we have the cell wall. What about the cell wall depositions? The cell wall depositions such as lignin and everything. In this case, the cell wall, the last stage will happen is, cell wall deposition will start. The cell wall depositions will start now. Cell wall depositions will start. Cell wall depositions will start. The first phase of your meristematic phase, where the cells are divided rapidly. The second phase of your growth is your elongation phase. The second phase where the cells are getting elongated, there is increase in size of the cell. Then we have vacuoles. The vacuoles will start to occur. Then we also have the secondary cell wall here, which will just start to appear. And in the secondary cell wall, listen to me very carefully, the depositions will start to occur. Those are the properties, those are the characters of your elongation phase. Clear? Any doubt here? Any doubt here? Vacuoles will occur, that is, vacuoles are appearing now. Vacuoles are appearing. Any doubt here? I'll go to the last phase of your growth, that is your maturation phase, also called as maturation phase. The first point is very simple. It is the third phase. Third and the final phase. Now in this final phase, the cells are maturing. Means the cells are differentiating here. The cells are what? Differentiating. The cells are differentiating. Yes, cells are differentiating. There are two main changes here which is happening. The first main change is in the cell wall. The first main change here is in the cell wall. The cell wall here will have complete depositions. The cell wall here will have what? Complete lignin depositions, or it could be pectin in some cases. Pectin in some cases, that is, lignins are depositing now. Clear? Such as your certain phloem. The next major change in the case of your maturation phase is nothing but your change in the protoplast. Change in protoplast. What are the changes in protoplast? Your protoplast, the vacuoles, right? The vacuoles will become maximum. Vacuoles will become maximum size. The vacuoles will become maximum size, and all the cells will have a definite structure and a definite function. All the cells here now will have what? Definite structure and a definite function. Definite function. That is the end of your maturation phase of the growth of your plant. So first part was what? The first part was your meristem phase. First phase, meristematic region, such as your SAM and RAM. We have small cells, dense cytoplasm, dense protoplast. Vacuoles are just absent. We don't have vacuoles, just starting stage. Primary cell wall, yes. And we have what? Multiple plasmodesmatal connections in your meristematic phase. Then we have your elongation phase. Second phase, where your cell size is increasing, length is increasing, cell size is increasing. Vacuoles will start to appear now. Yes, vacuoles will start to appear. Secondary cell wall formation happens, and along with that, in the secondary cell wall, the depositions will start to occur. But the final depositions, that is complete lignified cell wall, will happen only in the case of your maturation phase, that is completely cell is differentiated. The cell wall is completely lignin deposition. There are certain changes in the protoplast, such as the vacuoles will attain the maximum size, and each and every cell at the end of your maturation phase, each and every cell will have a definite structure and definite function. Fixed structure. Clear? In maturation phase, what happens is the cells will start to differentiate. Differentiation, they are becoming mature. They are realizing their role. Every single cell now has a fixed role as well as a fixed structure. Fixed role as a fixed structure and a fixed function. Change in protoplasm, that is vacuoles in your maturation phase will become maximum. In the case of your maturation phase, right, in the cell wall will have complete depositions. Clear? Any doubt here? Ask me right now, because I will show you diagrammatically now. I will show you diagrammatically how it happens. Ready? If this is your soil, this is the soil. From the soil, what grows? Inside the soil we have the roots. Yes sir, inside the soil we have the long taproot. And then we have certain root hair also. Now I told you the cells, the dividing cells are found at the RAM on SAM. So in this particular root here, which is growing, meristematic region can be found in your this part here, which shows meristematic region. Meristematic region, yes, meristematic region, where the cells are actively dividing, small cells, dense cytoplasm, no best primary cell wall here. Now here can you see this region? It's a much more elongated region. This is the region of elongation. Yes, region of elongation, where cells are elongating, cells are increasing in length, cells are increasing in size. This region here is region of your maturation. Region of your maturation, where cells have secondary cell wall, complete deposition, cells are completely differentiated, cells have a fixed role and a fixed size. Clear? Now here, I want all of you to understand here is, from the soil, taking soil as a reference point, this region, the most outside part, the furthest part is called as your distal region. Yes, it's called as distal region. The closest region, the closest to the soil, is called as proximal region. Now students, if you read your NCERT is clearly telling a line. What is it? Taking this, now let's understand elongation now. The region, you tell me in the chat now, the region which is distal to the elongation is what? The region which is found distal to elongation is meristematic region. The region which is found proximal to elongation is maturation. Clear? Students, remember, taking soil as a reference, the region closest to the soil. I repeat again, the region which is closest to the soil is called as proximal region. The region which is called as furthest to the soil is called as distal region. Now I'll ask you one more question. Which is the distal region? Which is the distal region to your region of maturation? The answer should be elongation. This is more distal to this. Similarly, the region which is proximal, listen to me very carefully, the region which is proximal to your meristematic is, proximal is, the closest is your elongation. Clear? Every single region has a proximal region and a distal region. Distal means the farthest end, proximal means the closest end. Clear? Tell me in the chat right now if this point is clear. I will show you NCERT diagram now. If you look at NCERT diagram right now here, that is 15.3 diagram. This is your root elongation. The radical is growing. The radical is growing, the root is growing. Can you see this tree here? This tree is nothing but your region of your maturation. This is your elongation. And this, can you see the last part here? This is the most dividing cells, small cells, dense cytoplasm. This is the region of meristematic. Meristematic. See, detection of zones of the elongation by parallel lines of technique. This is parallel lines of techniques. Different zones they are telling you. Different zones they are telling you in your NCERT. Clear? I hope this point is also clear to all of you, because next we'll be starting with the next concept. That is your growth rate. Can we start? Can we start with the growth rate? Now, what is growth rate? Growth rate, that is nothing but at what rate the growth is happening. Yes, growth rate is nothing but at, let me write down the definition here. What is growth rate? What is the meaning of growth rate? Growth rate is nothing but increase in growth, increase in growth per unit time, at what particular time, at how much time the growth is taking place. Now, depending upon your growth time requirement, we have two types of growth here. Okay, in your NCERT they have given you two types of growth. So students, the first type of growth I want all of you to notice, write down as your arithmetic growth. The first type of growth is arithmetic growth. The first type is called as what? Arithmetic growth. First type is called as arithmetic growth, and second one is called as geometric growth. Geometric growth. So two type of growths. One is called as arithmetic growth. The second one is called as your geometric growth. Students, I'll draw the diagram. I'll tell you the difference. So please write down right now. I want all of you to please mention this. Write on your book. As I'm drawing here, please write down in the similar fashion. Okay. Now, what happens in arithmetic growth? Listen very carefully. In arithmetic growth, we have a single cell. Yes, sir, we have a single cell. Now, this is a meristematic cell. Yes, this is a meristematic cells. All of us know the meristematic cell will divide. Yes, this particular cell right here will divide into two daughter cells. But one of the two cells will become permanent. This will become what? Permanent cell. This will become a permanent cell. Means what? This particular cell here will lose. It will lose its ability to divide completely. Listen to me very carefully. In arithmetic growth, one meristematic cell will divide into two. Out of the two daughter cells, one cell will keep on dividing. The other cell will become permanent. the other cell will become completely permanent. It will lose all its ability to divide. Similarly, now if this cell divides, one of the cell will be meristematic again. The other cell will become permanent. This is coming as such permanent cell. See, out of the two cells, one is becoming meristematic, other one is becoming permanent. Similarly, if you divide again, one cell will remain meristematic, the other cell will become permanent completely. Other cell will become completely permanent. So what do we understand by this? What do we understand by this diagram? What we understand here is, out of the two daughter cells, I'll write here. Out of two daughter cells, one remains meristematic, one remains meristematic. The other cell becomes permanent. That is the meaning of your arithmetic growth. Clear? In the case of your arithmetic growth, one cell divides into two cells. Out of the two cells, one cell remains meristematic, the other cell becomes a permanent cell. Again, this cell divides into two. One cell completely becomes meristematic, the other cell is again permanent. Similarly, again, if this daughter cells divide, one cell will become meristematic, other cell will become permanent. That is your meristematic, that is your arithmetic growth. Now let's understand your geometric growth. Now geometric growth is very simple. Very simple. One cell is meristematic. It is dividing into two cells. In the case of geometric cell, both cells, I repeat again, in the case of geometric, both the cells becomes meristematic. There is no permanent cell formation here. In the case of your geometric, both the cells become meristematic in nature. Clear? Both cells become meristematic in nature. Now can you see this type of growth? This type of rapid growth is called as exponential growth. Exponential growth. Because in this case, right, in the geometric way, we do not have any permanent cell. So cells are keep on dividing, the cells will keep on dividing. So there will be a complete exponential rapid growth. A complete rapid growth can be seen in the case of geometric growth. Now this is seen in the case, what happens here? Both daughter cells are meristematic. Both daughter cells are meristematic. Both daughter cells are meristematic in nature. Clear? Now amazing. Now I'll tell you the graph. And this is where it is more important. The graph is more important. Students, please draw with me. Because this can get very confusing very fast. Because some of the students will be like, no sir, I don't like maths. Why do I have to study maths in biology? In fact, why do I have to study maths in botany? But remember students, in your ecology you have to study graphs, right? In your botany also you study maths. So draw with me the graphs now. All of you, draw with me the graphs for your arithmetic first. X-axis, Y-axis. Let's take X-axis time. Yes sir, let's take X-axis time. The Y-axis, let's take length. In the case of your arithmetic growth, always remember it doesn't start from zero. No sir, it doesn't start from zero. In the case of your arithmetic growth, there is always a linear graph from here. Always a linear graph. Always a linear graph. And we have a formula here. Formula can be asked. It can come as a labeling or as well as a missing the formula. The formula here is LT equals LT equals L naught plus RT. LT equals L naught plus RT. Now what is LT here, students? The LT here is length at time t. The LT here is length at time t. But can I also call it final growth? It is very easy to remember. The final growth of a cell, the final growth of a plant, or final growth. Yes, LT can be written as length at time t or final growth. Now what about L naught? The L naught is again length at time 0. Length at time 0. Can I also call it initial growth? Yes sir, can I call it initial growth? You can call it as initial growth. Now what about your RT? This is plus. Now what about R? R here is nothing but your growth rate. The R here is nothing but your growth rate, also called as your elongation per unit time. If you open your NCERT, it also says elongation per unit time. Elongation per unit time. Now what is T again? T is, all of you know, T is time. T is nothing but your time. So students, in the case of your arithmetic growth, we have a linear graph. That is a constant growth is there. Yes, there is a very constant growth. Can I also call it a constant growth? Yes sir, there is a very constant growth is happening here. That is, we find a linear graph. When you plot between time and length, we get a linear graph. In that we have a formula. In that, students, we have a formula that is LT equals L naught plus RT. What is LT? LT is your length at time t, or also called as your final growth. Also we have L naught. L naught is nothing but your length at time 0, called as initial growth, the starting growth. Then we have R, that is growth rate. R can be told as growth rate or elongation per unit time. And finally the T here is called as time. Now the next question which will be always asked is, what is the example for this, sir? Where do we find arithmetic growth? The answer is very simple. This can be seen at, this particular type of arithmetic can be seen in case of your root growth, where in the case of your root elongation, in the case of your, what is the example? Root elongation. Root elongation at constant rate, at constant rate is the example here. Example is your root elongation at a constant rate. Students, remember, this is the information which all of you to write down, all of you should remember for your arithmetic type of growth. What is arithmetic growth? In case of your arithmetic growth, one daughter cells will divide, other daughter cells will remain permanent. Clear? Now let's understand geometric growth. Write down with me, all of you. All of you write down with me. In case of your geometric growth, also let's draw a graph. Yes sir, let's draw a graph. Again, x-axis, y-axis, x-axis and y-axis. Here x-axis is again going to be your time. The y-axis is going to be weight. Y-axis is going to be weight. Now students, write down with me very carefully now. This is where we get not a linear graph. In the case of your geometric growth, we get a sigmoid curve. What do we get? We get a sigmoid curve. What is sigmoid curve? This. If you write down a sigmoid curve, it looks something like this. Now, in this sigmoid curve, when the plant is initially growing, it requires time and preparation. Yes, it requires time and preparation. In case of your geometric growth, the initial phase is very slow. In the case of your geometric growth, the initial phase is very slow and we call it lag phase. What do we call it? We call it lag phase. Lag phase. But as soon as the lag phase is over, your plant cell is completely ready to grow exponentially. It is ready to rapid fire. It is ready to take off and blast. We get the exponential phase, or also called as your log phase. Can you see this part? Exponential phase or log phase. Log phase, also called as exponential phase. Exponential phase. Now, once the plant is completely grown, but the nutrients are reducing, the nutrients are decreasing very rapidly. Nutrients are decreasing. The plant reaches a stage called as stationary phase. Finally, the plant reaches a stage called as stationary phase. Can you see this part here? This is called as what? Your stationary phase. Why? Because less nutrients. Because of the less nutrients, because of lack of nutrients, there's a decrease in the growth called as your stationary phase or a decline phase, also called as your decline phase. Now, can all of us write down the formula for this? The formula for this is WT. WT equals your W naught e to the power RT. The formula for this geometric growth is WT equals W naught e to the power, that is log of your natural logarithms, e to the power of RT. Can I tell again here? WT is what? What is WT? Similarly, same here. Weight at the time t now. Weight at time t. Can I also call it final weight? Yes sir, we can call it final weight. Now what about W naught? W naught is nothing but your weight at time 0. At time 0. Can I also call it initial weight? Initial weight. Yes sir, I can call it initial weight. Now what is R here? The R here is nothing but your relative growth. NCERT is telling you growth rate, you can also write that, but better understanding, I'll tell you why. I'll tell you why should be relative growth in our next few minutes. Relative growth rate. Relative growth rate. Again, your T is your time. T is your time. Students, this is the graph for your arithmetic, which was a constant linear graph. Yes, a constant growth is there, and the formula is LT is equal to L naught plus RT. But for your geometric growth has 3 phases. Initially, it is a very slow growth, that is called as your lag phase. But as soon as all the nutrients are there, it is ready to blast. That is called as your exponential phase or log phase. Then as soon as all the nutrients are over, it reaches a stage called as your stationary phase. We have a formula here, that is WT equals W naught e to the power RT. What is W naught? W naught is your initial weight, WT is your final weight, E is the natural logarithm, R is relative growth rate, and T is your time. T is your time, and this is the growth rates. 2 types, arithmetic as well as geometric growth rate. Now students, one example is very famous example here is, very famous example for your geometric is the division of your zygote. Division of zygote. Very important here. Division of the zygote. If this is the zygote will divide and become the embryo. Yes sir, zygote will divide and become the embryo. Students, remember this very carefully, very tricky thing. Starting, initial growth of the zygote is always geometric. It is always geometric. But later on, when it forms the embryo, later on it switches to arithmetic. It switches to arithmetic growth. Switches to arithmetic growth. Initially, zygote will divide geometrically. Then, after the embryo formation, it divides arithmetically. Clear? Example is clear? Any doubt here? Students, this is a very easy chapter. You should understand every line of it very carefully. You will love this chapter. Okay? Understood? Can I tell you NCERT diagrams? Let's not leave any NCERT diagram also. See, this is the arithmetic. Can you see here? One is remaining meristematic. The other one is becoming what? Permanent. Similarly, one is remaining meristematic. The other one is becoming permanent. This is coming from here. Similarly. Again, the next one is one remains meristematic. The other one becomes permanent. Meristematic, permanent. This is arithmetic growth. But in the case of your geometric, each and every daughter cells will remain meristematic in nature. Each and every cell will remain meristematic in nature. See here, this is for your zygote. Initially, the zygote will show what? Initially, the zygote will show geometric growth. Yes, initially, zygote will show geometric growth. But once it forms here, from here onwards, it shows arithmetic phase. Later on, once embryo formation happens, it switches to your arithmetic phase, where one remains meristematic, other becomes permanent. That's why we know even in human, remember, one zygote formation embryo happens. Later on, what is the meaning of permanent? Means that it has a very fixed function. As the embryo is growing, we have specific organs, specific tissues performing specific function later on. That is what is happening here. That is initially capable of dividing. Later on, we have some cells becoming very permanent by losing its ability to divide. Clear? See here, this is your arithmetic graph. Starting from here, linear graph. But in the case of your geometric, we have the lag phase, initial, where the preparation is happening. Then we have the exponential, also called as log phase. Then we have only the stationary phase, which is coming here, and this right here is a sigmoid curve. Idealism of sigmoid growth, which is seen in the case of your higher plants. And most of the cells in the plant's body, almost all cells divide by your geometrically. Almost all the cells divide geometrically. Remember that. The example for arithmetic growth is the root elongation. The root elongation is the example for your arithmetic growth. Because why? It is constant. It is constant linear growth. Students, a question will come from here. Remember, your arithmetic growth is constant always. Remember that. Okay? Next, ways to measure growth. Can we start? Last one small topic is here. What are different ways we can measure the growth? What are different methods? The answer is two types. What are two types? There is something called as absolute measurement. One is called as relative measurement. Can we write down? Please students, write down with me. These are the notes which will help you. These are the notes which will actually help you, which I am preparing for you live. Please write down with me all these notes, and after this, just read your NCERT. Just read your NCERT once, everything will be set. This is the 90% of studying you're doing right now. 90% studying you're doing with me. 5% is your NCERT, 5% is your question solving. That's all. The chapter is set. Okay. Now students, the growth are of two types. Ways to measure is two types. Ways to measure. Different ways to measure, there are two types. The first one is called as your absolute growth. Absolute growth. The other one is called as relative growth. Other one is called as what? Relative growth. Now, what is the meaning of absolute growth? I'll give you a simple example. I was 60 kgs last year. Now I am 65 kgs. What do? Final minus initial. 65 minus 60, we get 5 kgs. So I have increased by 5 kgs. Simple. Yes sir, it is very simple. that is your absolute growth. That is your absolute growth. That is final minus initial growth, we get the absolute growth. For example, we take someone from your chat. Let's take Crazy only. Crazy is right now, let's say 60 kgs. Last year, your Crazy was 50 kgs. How many kgs growth did he see? He saw of in total 10 kgs growth. Final minus initial, we get the growth. That is your absolute growth. But students, remember, what is relative growth? Relative growth is always and always in terms of percentage. In terms of percentage. Let me give you an example. Let me give you an example. Let's say Ravindrani is there. Okay. She is getting, her parents are very strict. They're like, I will not give you money. You should study right now. You should only study. Money is not concern for you. I will give you pocket money of 5 rupees per month. Okay. Just 5 rupees. 5 rupees you can take, go buy a chocolate every month and eat. Okay. 5 rupees pocket money Ravindrani is getting. But next, after 6 months, Ravindrani is fighting in the house. No, I want more money. So your pocket money has increased from 5 rupees to 10 rupees. Yes, from 5 rupees to 10 rupees. Meaning what? Your pocket money became double. Can you repeat? Can you see that? From 5 rupees to 10 rupees. There is complete 100% increase. 100% has increased. 5 rupees to 10%. 5 rupees to 10 rupees. That is 5 rupees increases happened. Now let's take one more person. 5 rupees. Let's take, who should I take? Who should we take from the chat? Anyone from chat who wants to volunteer for the next question? Let's take Sanvi. Sanvi's pocket money is 50 rupees per month. Sanvi's pocket money is 50 rupees per month. She has fought so much in the house that her pocket money has become 50 rupees. Now students, her pocket money is also increasing by 5 rupees. From 5 rupees, her pocket money became 55 rupees. In both the cases, we have 5 rupees increase. But who is more happy here? Who can tell me in the chat? Who is more happy in the chat? Is it your Ravindrani or is it Sarvi? Remember, Ravindrani's pocket money became 100% increase. But in the case of your Sanvi, it was just 10% increase. Just 10% increase. Now, this type of growth which we see in percentage, is called as your relative growth. It is called as what? Relative growth, which we see in percentage. Even though in both the cases, the pocket money is increasing just by 5%. Sorry, 5 rupees. But percentage wise, Ravindrani's pocket money became 100% increase, while your Sanvi's was just 10%. Just 10%. Now let me write the formula for all of you. Then I'll show you same example with your NCERT. Okay, write down. In the case of your absolute, we always do final minus initial. Yes, we do final minus initial. And this is called as what? Final minus initial. Example, your weight was 60 kgs. Final, initial was 50 kgs. In growth is just 10 kg. What about relative growth? In relative growth, we do final minus initial divided by initial into 100. Into 100 when you do, you get the answer in percentage. You get the answer in percentage. Now let's look at the example. Let's understand this better with the help of example. Let's go with an NCERT example. Remember the formula for your absolute growth? It is final minus initial. That is absolute growth. But for your relative growth, it's always final minus initial divided by initial. We get the relative growth. Can we start? NCERT example. Now, NCERT is giving a beautiful example, just like your pocket money. What is the example? We have two leaves here. We have two leaves here. We have leaf number A, leaf number B. Leaf number A is your Ravindrani. Okay. Initially it was just 5 centimeter. The leaf was just 5 centimeter. But the leaf later became how much? 10 centimeter. Look at the leaf number B. It was 50 centimeter square initially. Then it grew. The surface area increased to 55 centimeter square. Write down with me the absolute and the write down with me the absolute as well as the relative growth for your leaf A. For your leaf A, what is your absolute growth? What is your absolute growth? For your leaf A, absolute growth. For your leaf A, the absolute growth will be how much? What is final minus initial? 10 minus 5. Final minus initial. You do 10 minus 5, how much you get? 5 centimeter square. That is the absolute growth. What about your leaf B? What is the absolute growth? For your leaf B, what is the final? Final is 55. 55 minus 50, how much is it again? 5 centimeter square. Yes, it is 5 centimeter square. Now let's do relative growth. Let's do relative growth. What is the relative growth? Final minus initial divided by initial. Yes, this will be how much? 5 by 5. That's again 100%. 100% increase. In the case of your leaf B, the relative growth will be how much? What is the final? 55 minus 50 divided by 50, which will come up to just 10%. Just 10% increase. So this is the different ways to measure the growth. We have something called as your absolute growth and we have something called as relative growth, which we can measure the growth in. Clear? Is the concept clear? Is the concept clear to all of you? Or do all of you know what is the difference between your absolute growth as well as relative growth with the example from your NCERT? Clear? Now, let's try to do a question. Let's try to do a question first. Can we do a question? What is the question? A typical growth curve in plant. A typical growth curve. Is it linear? Is it zigzag? Or is it parabolic? Or is it sigmoid? Which is it? A typical growth. A typical growth in the case of your... Sir, from this numericals will be asked. No. But you should know what is absolute growth, what is relative growth. Okay. The answer is sigmoid. The almost typical growth is always your sigmoid, but in the case of root, it is linear. Next question is here. Next question. Which of the following statements are correct regarding the type of growth represented by the diagram? So we have a diagram here. Let's understand the diagram first. Students, you tell me in the chat. Is the diagram arithmetic growth or is it geometric growth? Yes sir, we can see there is no sigmoid curve. It is a linear graph. It is arithmetic growth. Yes, it is an arithmetic growth. Now let's read this. It is observed that initial stage of the embryonic cell division. Do we find this in the embryonic cell division? Initial stage. Remember I told you, in the embryonic stage, the initial is geometric. Then we have arithmetic. Growth rate is constant. It represents the root growth tip. It is represented by the expression L1 is equal to L0 or LT is equal to L0 e to the power of RT. How many statements are correct here? 1 and 3 are correct, sir. Only 1 and 3 are correct. 2, 3 and 4 are correct. 1, 2 and 4 are correct. Only 2 and 3 are correct. 10 seconds. Time starts now. 10 seconds. Your time starts now. Fast. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Time's up. That is, the first statement is obviously wrong, students. We know in the development of the zygote, the first part is geometric, then it is arithmetic. Wrong statement. Yes, the growth rate is constant here. Constant growth. Found. Where is it found? It is represented by the root growth at tips. Yes, the root elongation. Is it the expression for it? No. This expression is for your geometric growth. So answer is very simple. The answer is your statement second and third are absolutely correct. Now, the next part of your chapter. The next part of the chapter is conditions of growth. When the plant is growing, what are the requirements for its growth? What are the requirements for the growth? The requirement for the growth is, yes, the plant will need optimum temperature to growth. Yes, it needs optimum temperature. Then it needs nutritions. Yes, it needs nutritions. Various type of nutritions for the plant to grow. It needs light. It needs water, and it also needs oxygen. It needs oxygen. But what is the specific requirement? I'll tell you now the specific requirements of each one of them, which is also given in your NCERT. Write down with me, students. So if you notice, I am writing more so that all of you can make notes with me. So that is the reason I'm writing more. So all of you can also write down with me. Sir, what is efficiency index? Efficiency index is not in your syllabus, so please don't study it. Okay, I am sticking to NCERT. They will not ask you efficiency index. Okay, efficiency index would have come from your this thing, absolute and relative growth. Don't worry about that. Okay, so let's understand conditions for growth. The first one is your water. Now, why does the water requirement? Where is the water requirement? The water requirement is very simple. The turgidity. The cells, the stomatal cells or any cells will absorb the water and they become very turgid. Yes, they become very turgid. So they require turgidity. Yes, can I write turgidity? That is, the cells will swell up. Yes, the cells will swell up. Turgidity. The other requirement for water is all the enzymatic actions need a medium, right? They need a medium to work. The medium for enzymatic action is your water. It is a medium for enzymatic action. Yes, it is a medium for enzymatic action. Now, everyone should remember this turgidity is nothing but your extensive growth. There will be extensive growth. Now, the second one is your oxygen. Now, you should tell me, why do plants require oxygen? Anyone in the chat fast? Last chapter, we just learned plants need oxygen for aerobic respiration. Plants require oxygen for metabolic process such as your aerobic respiration. Yes, they need for aerobic respiration. Yes, they require for aerobic respiration. The next one is nutrients. The nutrients such as your macronutrients as well as micronutrients. You are so lucky and also unfortunate at the same time, because this chapter mineral nutrition is so important, because that chapter is actually related to many chapters. So you won't be studying this part here in detail. So I'll just write that nutrients can be macro and micro. What are they needed? For synthesis of protoplast. Synthesis of your protoplast or protoplasm, not protoplast. There is no cell membrane required, so protoplasm. And also in some cases this provides energy, also for your energy requirements. The next point is your temperature. Yes, plants will work at a very specific temperature. Yes, everyone has an optimum temperature. You, me, all of us, we have a 27 degrees where we work the best. Similarly, plants will have an optimum temperature. Plants will have a optimum temperature where Pacific work at. The next one is your plants require light. Yes, they require light for photosynthesis. The other one is your plants, that is conditions for growth. Is again, gravity is also very important. Gravity is also very important. For example, geotropism. The roots will grow towards the gravity. The shoot will go against the gravity. So these are the different 6 points which you need to know for conditions for growth. Okay. That is the first part of the chapter is done. First part of the chapter is done. Now I want all of you to understand the next part of the chapter is, this is very important where so many students still get confused. I've been teaching this part from very long time. That is, what is the difference between differentiation, de-differentiation and redifferentiation. Ready? Students, please write down this one. After today's class, you will never forget what is the difference between differentiation, de-differentiation and redifferentiation. Ready? Ready with me, all of you? I want green hearts in the chat today, because today is for botany. Today we are doing last part of your last chapter for your plant physiology. So I want to see a lot of green hearts in the chat. We will start the next part, that is your differentiation, de-differentiation and redifferentiation. So first I will make you write down this, then I will explain you with the example. Okay? First I'll write down this, then I'll tell you an example. Now remember, we have one cell. Think here. First we'll do differentiation. First we will do differentiation. Okay? Let's write down with me. First we'll start with differentiation. Now, first we'll do differentiation. Now imagine we have one cell. Now this particular one cell is meristematic in nature. This one cell here is meristematic in nature. One cell. It's meristematic in nature. Now you tell me in the chat, does the meristematic cell has ability to divide? Yes sir, meristematic cells can keep on dividing. They have dense cytoplasm, small size, conspicuous nucleus, primary cell wall and everything. Yes, same points. So this particular meristematic cell will divide. But at one point, this meristematic cell will lose its ability to divide. It will lose its ability to divide. This meristematic cell will lose ability to divide. I'll tell you a small story now. Some of the students after class 12, because of some financial conditions, all of you are so lucky. All of you are so lucky that you have education with you. You are preparing for NEET examination. But there are some students who after 12th, they will start working because of some financial conditions in the house. They don't have money. These are those cells. These are the sad cells which has so much on their plate right now. They cannot study anymore. They have to start working after class 12th. Just after class 12th, they will start working. That is, these are the meristematic students which will lose ability to divide. That is, they will stop studying and they get a permanent job. They become differentiated cells. They become differentiated cell. That is, they become differentiated cells and they find a job. That is, they become primary permanent cell. They become primary permanent cell, or can I tell primary permanent tissues. The condition is so bad sometimes that they become primary permanent tissue just after class 12th. Now what are the examples here in terms of botany? In terms of botany, I'll tell you a few examples here. Primary xylem. These are students. Primary phloem is a student. Primary phloem. As well as your primary medullary race. Primary medullary race. This is the story of differentiation. In differentiation, students, in case of differentiation, we have meristematic cells. Unfortunately, because of some condition, they cannot study further. That is, they lose their ability to divide. They become differentiated cell and they find a job. They find a job after class 12. That is, they become primary permanent cell or tissue. Primary permanent cell or tissue. That is, they've become primary xylem, primary phloem or medullary race. That is the story of your differentiation. Now students, listen to me very carefully. Listen to me very, very carefully. After some years, they become families all right now. Then they decide, no, I will actually start studying again. I will do a BSc course. I will do a course now. I will join a degree. I want to finish my studies. The fire inside them comes back again. The fire inside them comes back again. That is a differentiated cell. This was your permanent cell. Permanent cell will again regain the ability. After some years, that particular student, when the family is all right, it will start to divide again. That is, it regains the capacity. It regains the capacity to divide. It means particular student is able to study again. And once it regains the capacity to divide, it becomes secondary meristem. Secondary meristem. It becomes what? Secondary meristem. This is called as what? This is called as de-differentiation. That a particular permanent cell which had lost everything, which had lost its ability to divide, again after some time, it regains the capacity to divide. The student is able to study again, and it becomes your secondary meristem. Students, remember, this only happens in the case of your dicots. It only happens in the case of your dicots. Example, remember the student here. Primary meristem. This particular primary meristem. One second. This particular primary meristem will now become this primary meristem. One second. Here. Sorry. Remember this medullary race. This particular student here. This medullary race will differentiate. This primary medullary race. This primary medullary ray will divide. This primary medullary ray will divide and becomes your vascular cambium. Becomes vascular cambium. This is the story of your de-differentiation. This is the story of your de-differentiation. So remember students, in the case of de-differentiation, the permanent cell will regain the capacity to divide and it forms the secondary meristem. Now you tell me, this particular secondary meristem, can it divide? Yes, meristematic cells can keep on dividing. That's why they're called as meristematic cells. Now this student here, he gets his BSc degree, but he always had a dream. That he wants to become a musician. Yes, he didn't want to study the study. The studying only came because he wanted to earn more money. Just because he wanted to earn more money, he did a BSc degree. But now, when everything is alright, he wants to pursue his own dream. What is it? He wants to become a musician. That is the third part of your story, that is redifferentiation. The third part of your story is redifferentiation, that this particular secondary meristem loses its ability to divide. And it performs specific function. It performs a very specific function. That is, now this particular student performs a very specific function, that is, he becomes a musician. This is called as, and it forms the secondary tissue. It forms the secondary tissue. This particular vascular cambium divides and forms the secondary xylem, secondary phloem. Students, listen to me very carefully from starting one more time. We have a student who's studying in class 12th right now. He finishes class 12th. We have your meristematic cell, which is your student, but due to financial condition, the student is not studying further. He starts to work. That is, the meristematic cells lose ability to divide. It becomes a differentiated cell. Now this cell becomes permanent cell. Yes, it becomes a permanent cell. So this is called as what? Differentiation. The meristematic cell is differentiated to become a primary permanent cell. Now this primary permanent cell, what happens now? It regains its capacity to divide. After some years, the student is studying again. The student is doing a BSc degree again. That is, regains the ability to divide, and it forms the secondary meristem. It forms the secondary meristem. This is called as dedifferentiation. Now from here to here, now this student here, he decides, no, I do not want to become any type of education point. He doesn't want to do BSc anymore. He wants to follow his true passion. The true passion was what? The true passion was musician. That is, it loses again its ability to divide, and it performs a very specific function, that is secondary tissue. This is called as your redifferentiation. Redifferentiation. So what is dedifferentiation? Dedifferentiation is again regaining the ability, becoming secondary meristem. The secondary meristem again is losing all its ability, losing its power to differentiate and performing a very specific function, becoming secondary tissue. This only happens in the case of your dicots, that is, vascular cambium becomes secondary xylem. Vascular cambium becomes secondary xylem, and your secondary phloem as such. Clear? Clear? Students, I hope everyone remembers the story of differentiation, dedifferentiation and redifferentiation. Please write down this, because in the examination they will never ask you the story. They will ask you this in the examination. So please write down. Now the questions will come from where? I'll tell you. The questions will come from your examples of differentiation, examples of dedifferentiation, examples of your redifferentiation. Those are the examples which will come in your examination. Clear? Amazing. Respiration PDF I'll send today. Respiration PDF I'll send today, okay? Respiration PDF I will send you today. Now, can we start with development? Can we start with development, everyone? Can we start with development? Can we start with development? Let me know in the chat if I can start development. That is, the growth part is over now. Growth path is done. You did so many concepts, such as different ways to measure growth, types of growth, arithmetic growth, as well as your geometric growth, your absolute growth, relative growth. Every type of growth which is given in your NCERT is done and dusted. Every single point has been covered till now. Open your NCERT, read once again. You will see, yes sir, you have covered all the points. Now students, it's time for your development. Time for your development. Now I'll tell you basic difference, basic definition. What is a development? Development is nothing but all the changes which are happening in a plant's life cycle till death. Write down with me. All the changes, Now these changes are in a sequence. Remember, all these changes are in a particular sequence. All the changes that occur in plant's life cycle. Yes, all the events, all the different types of events, developmental paths, developmental events happening in a plant's life cycle is called as development. Now for example, we have a seed. The seed will undergo what? The seed will germinate. I told you what is germination. Now this seed will undergo germination and produce a small seedling. Germination. This is your seedling. Now after this seedling will develop. This seedling will develop into a small, like a protonema stage, like a juvenile stage. Yes, juvenile stage. Yes, it will enter a juvenile stage. Juvenile stage. Now students, remember, in this juvenile stage here, it cannot do reproduction, because in the juvenile stage only vegetative parts, only and only the vegetative parts are developing. Only and only the vegetative parts are developing. Now this juvenile phase will become mature. Yes, it will reach in maturity, that is sexual. Once it attains maturity, the reproductive organs will develop. Yes, reproductive organs will develop. Now once the maturation happens, what happens? After maturation, if the reproductive parts are there, can it do flowering? Yes, flowering will happen. Yes students, flowering will happen. Now once flowering happens, what happens? Then it is becoming old. Old age is happening. We call the old age in the case of plants senescence. In the case of plants, we call it senescence. Now once senescence is happening, finally there will be death of the plant. Finally there will be death of the plant. Why am I laughing? Because suddenly I saw so many green hearts. I was overwhelmed. I'm like, oh, so many green hearts. Okay, that's why I was laughing. So what is development? Development is nothing but all the changes that occur in the plant life cycle. It is a sequence of events. Yes sir, it is a sequence of events. For example, we have the seed which is germinating. We get a seedling. After seedling, we get juvenile stage. In the juvenile stage, there is only and only what? Vegetative parts. Now this vegetative parts becomes mature. That is, we get reproductive organs. Once the reproductive organs are developed, yes, once the reproductive organs are developed, we have flowering. Now once the flower develops, beautiful flower, what happens? Reproduction happens, pollination happens. After that, the plant is becoming old, that is senescence. Now after senescence, we have the death of the plant. This is the complete life cycle, not only for plant, even for all of us, except the flowering. Except the flowering, we have the same pattern. Except the flowering, we have the same pattern. Look here, your NCERT is also the same. Your NCERT also is the same. That is, NCERT is telling you a plant is cell is dividing. We have the meristematic cells. That is a dividing cells. Now this particular dividing cells, plasmatic growth will happen. What is plasmatic growth? Growth in the protoplasm. What is plasmatic growth? Growth in the protoplasm. Growth in the protoplasm. Yes. Then there is elongation. Yes, all of you know what is elongation region, elongation zone. Then we have differentiation, maturation, and we have a mature cell. Finally, we have a mature cell. And this particular mature cell will develop, will produce your flowers, will produce your reproductive organs. Then we have the senescence, and finally the death. What is this? The sequence of developmental process in plant cell. Every single cell will go through this particular stage in the case of your plant. In the case of your plant. Clear? Class duration, probably I'll do 1 more hour and we'll stop after 1 hour. The class will be over after 1 hour. Chapter is over. Yes, until the chapter is over. Old age means senescence. Abscission means the leaf is falling down, is its abscission. Okay. Now, next concept which came in your NEET PYQ. This concept as a NEET PYQ, it came as a NEET PYQ. That is plasticity. What is the meaning of plasticity? Can we start? Can we start, students? I want everyone to like the video. I feel no one is liking the video. I see so many less likes. I see only 15 likes. What is this? Everyone like the video, and we'll start with the next concept, that is plasticity, which is your NEET PYQ. Okay. Now what is the meaning of plasticity? Now I told you students, I told all of you, plant has a particular path. Yes, green hearts. Plant has a particular path in its life. Yes, a plant has a fixed developmental pathway. Now, if a plant is going away from the path, it is changing its path. That is, if a plant is following, let me write it little up. If a plant is following a different pathway, what is the meaning of different pathway? It is actually undergoing change in different pathway, means change in developmental pathway. So development only change here. Developmental pathway. There is a complete change in a pathway. For example, it has to go from juvenile, it has to look like this. In adult, it has to look like this. But the plant is completely choosing a different pathway in its life cycle. That is called as plasticity. Plant is following a different pathway, that is a change in different pathway in response to requirements. In different response to requirements. Now, what are these requirements? For example, listen to me very carefully. For example, there are certain environmental factors. Not environment. First, let's type. NCERT is telling you first different thing. Let's follow NCERT completely. The NCERT is telling you the first one is, that is in response to your. One second, students, my legs are hurting. Where was I? A plant is following a different pathway, change in development pathway in response to different requirements. Now, what are these requirements? Requirements is that at certain stage or certain phase in life, there might be a certain different stage might completely change in the requirement because of certain change in the lifespan. The other one is nothing but your environment. The other one is different requirement in environment. There might be certain different changes in the environment, hence the pathway is changing. Or there might be a certain change because of the phase. For example, all of us know in your frog, when the frog is in the small stage, that is your tadpole. In the tadpole, the breathing is mainly through gills and skin, gills. But as the tadpole is becoming older, right, as the tadpole is growing, it develops a little, frog will develop a little lungs-like structure. That is, a frog will breathe through its skin as well as lungs. So as soon as the developmental stage changed, there was a certain pathway also was different. Similarly, even in the case of plants, there are certain changes based on which phase it is in. Example, I'll tell you, certain plants, when they are in juvenile phase, certain plants, when they are in your juvenile phase, certain plants, when they are in juvenile phase, the leaf will look like this. Certain plants, when they are in juvenile phase, the leaf will look like this. But as the plant is growing older, as the plant is going into the adult phase, the leaf will look completely different. Look at this. The leaf is changing completely. So the change in leaf, the change in pathway, how it's supposed to look like, is completely changed. Why? Because one is in juvenile phase, other one is adult phase. The example which is given in your NCERT for this one here is cotton, coriander. The other one is, let me think. Other one is your larkspur. Yes. Larkspur. Students, they can ask you a proper example in your NCERT, in your examination, they'll ask you. That is plasticity seen in the case of your cotton, coriander or larkspur. What type is it? It is actually heterophily. What is it? It is a type of heterophily. It is a type of heterophily. What is heterophily? Meaning of heterophily is simple, that is different types of leaf. Different kinds of leaf. Students, this has come as an question before. The heterophily, that is different kinds of leaf, which is seen in the case of your cotton, coriander, larkspur, is a type of change. It is a type of plasticity because of phase of life. In juvenile phase, it looks like this. And in the case of your adult phase, the leaf is completely changing its structure. That is your phase of life. The second one is environment. The second case is environment. The example is your buttercup. Buttercup. Now this, because of environment, I'll tell you simple. You tell me, use the logic. When the plant is going on in your air or terrestrial, or when the plant is growing in the land, the leaf looks like this somewhere. The leaf will look like somewhat like this. Yes, the leaf is looking like this somewhat. But when the same plant is growing in aquatic condition, it looks something like this. The plant's leaves looks something like this. In aquatic stage, in your aquatic, when the plant is in water, the leaf will look like this. Simple reason. The simple reason here is, if the plant did not change, did not modify, did not alter its pathway, if the leaf look like this in the plant, in aquatic, when the water is coming at high pace, this particular plant will be completely washed away. Yes, if the water is falling like this, it can create holes in the leaves. Yes, to avoid that, to avoid the pressure, to avoid the difficulties, the plant modifies its pathway. It modifies its developmental pathway and it becomes like this. The leaf of this particular buttercup in aquatic stage will look like this. So students, what is plasticity? Plasticity is a concept where we understand plant is following a different pathway. That is, a developmental pathway is completely changed in the case of plant in response to the phase in the life of the plant or the environment. For example, phase in life is your cotton, coriander and larkspur. Here, the plant leaf is different in the case of your juvenile phase, and in the case of adult phase, the leaf is completely different because of the phase development. Similarly, in the case of your buttercup, it is modifying itself. The leaf is changing its pathway, developmental pathway, because in the case of terrestrial, it looks like this. In the case of aquatic, it is looking like this. Why? To make sure there is a less resistance when the water is flowing. Clear? Clear? I hope this point is clear to all of you. See here, in juvenile phase, in cotton, coriander, larkspur, juvenile phase and adult phase. Similarly, in the case of your buttercup, terrestrial looks like this, but in the case of aquatic, it is becoming very thin like. Why? To make sure water can just pass through, there is no difficulty. Make sure there is no difficulty. Both of this, in the case of your phase of and also in the case of your environment, are example of heterophily. Both are example of heterophily. Example should remember cotton, coriander, larkspur, as well as buttercup. That is ranunculales. Clear? Any doubt here? Ask me first. Yes, this also can be a type of adaptation. Clear? Let me know in the chat by green hearts. All of you should spam green hearts in the chat if plasticity concept is clear to all of you. If plasticity concept is clear to all of you, just spam green hearts in the chat and I will go to the next concept, that is your development. What is development? Development is nothing but your growth plus differentiation. We just learnt it. Now this growth and development can be based on two different things. This entire development can depend on two type of factors. The entire development of the plant can be based on two factors. One is called as your extrinsic factor. One is called as your extrinsic factor. One is called as your intrinsic factor. Extrinsic factor and intrinsic factor. Now what is this extrinsic factor? Augustine is asking me, thorns can be considered an example. Yes, but not here. Thorns is also a type of modification, but this is based on your growth pattern is only changed. The complete growth pattern is changed here. Now in case of your extrinsic factor, all of you should know that we discussed already. Temperature, tell with me. Temperature, oxygen, water, nutrients, light and gravity. Yes, we learned this, sir. The different extrinsic factors done. Now what about intrinsic factor? In intrinsic factor we have two types again. Intracellular as well as intercellular. Intracellular as well as intercellular. Now students, all of you write down with me. In the case of intracellular comes the genetic factor. Comes the genetic factor. What does the mean of genetic factor? It is in your DNA, not your DNA or my DNA. It is in the plant's DNA that it has to follow a particular developmental stage. It is in the DNA. The information is in the DNA that a particular plant has to grow in this particular fashion. Juvenile phase, maturation phase, flowering phase. It cannot go ulta and all. It cannot, flowering cannot become first, maturation cannot become later. So it has a proper sequence which has to follow, that is intracellular in nature. It is genetic factors are involved. The other factor which we will be studying now, that is intercellular factor, is your plant growth regulators. The next concept which we will be studying right now is your plant growth regulators. Clear? Plant growth regulators. Start. I'll tell you a brief idea about plant growth regulators, but before that, can we take a 10 minutes break? I think we have been studying this chapter from past 2 hours. Yes, 5 to 6, 6 to 7. From past 2 hours, 15 minutes, we have started this chapter. Now let's take a 10 minutes break and we will start with the last part of the chapter, that is your plant hormones as well as PGR. Hopefully till now, students, if you do not have any doubt, till now in your growth and development, ask me any doubt right now. Write all the doubts here. I will come and clear all the doubts for all of you. Let's take a 10 minutes break and let's come back after 10 minutes. Okay? All of you go eat something, take some chips or eat some vegetables or something, or bring your dinner, drink some water. Let's meet after 10 minutes and we will start your hormones. Okay? Amazing. Before that, let me show you one slide which is important for plant growth. Not plant growth, this particular. Here. Before I go, let me tell you one small thing. That is your our Telegram channel. Students, you might have noticed on a Telegram channel, we have been uploading so many things just for all of you. That is, on a Telegram channel, we are taking weekly schedule is uploaded there. Yes, which teacher, what time, what day is going, Telegram channel. Daily quizzes, all of you know who are in Telegram channel, they know daily, every single day a PYQ comes for you. Then we have daily session PDF. After today's session, the PDF will be given to you on Telegram channel. Then we have weekly mock test, very important. The most important part. Start. Weekly mock test, the link will be given to you on Telegram. And you have Super 6 series complete playlist will be given to you, and which we are going to start now. Next, we are starting. The next we are starting is weekly live sessions on Telegram. Yes, you heard me right. On Telegram, I will be coming live and I'll be talking to all of you. No session, we are not teaching anything in there. Every single week, we'll be coming live on the Telegram channel. That is, we'll be just talking to all of you. Okay, let's take a 10 minutes break and we'll come. Someone Bala is asking me, seed germination is on. No. Seed germination, photoperiodism, and vernalization has been removed from your syllabus. Not there. 10 minutes break, let's go. Ten minutes break starts. Can we start, students? Where did we stop? So how is everyone doing? So can we start? So students, we started off with growth. I told you what is the meaning of growth, the types of growth, what is germination, what is development. Each and every concept is done and dusted. I hope till now you have understood each and every word and each and every concept. So students, from now onwards we'll be doing the second part of the chapter. So but this is the most important one. You can expect at least 2 questions from this particular part alone. That is, from the hormones alone, you can expect 2 questions this year, because that is the importance of this, the last part. So students, I want each and every one of you to please write down with me. But don't worry, I have a surprise for all of you. I have a surprise for all of you. What is the surprise? In the end of today's video, I will be giving you a proper table like this. I want all of you to write down this in your book, a proper table for each and every hormone function. Can you see? Every single thing is there in this table. So end of the video, we will summarize there. But until then, we will start with the hormones. So end of the video, I will give you a proper tabular column of each and every hormone function wise. So that's the place where question is going to be asked. The other part where the questions will come from, I will make you write it myself. So can we start? Can we start, everyone? I want green hearts in the chat so I can start. The last part of the chapter, less than 45 minutes, I'll finish it. Okay? So let's start. Don't worry, all the PDF will be given to you on the Telegram channel. So students, what are plant hormones? Similar to human hormones. These are the chemicals which control physiological process. These are the hormones which control physiological. Yes, they control physiological processes. What are the physiological process? The physiological process can be your division, it can be your cell division process or activity. Now this physiological process can be your cell division, right? It can be your cell division, cell differentiation. Yes, cell division, cell differentiation, or cell elongation. Yes, this can be cell differentiation, cell elongation, or your simple cell division. Now just like your human hormones, these hormones are also produced in your small quantity. That is, they are produced in your low concentration, very low concentration. Just like your human hormones, they are produced in very low concentration. That is, they are produced in your traces amount, so less, but it has a massive impact on the entire functioning of your plants. So students, the entire plant growth hormones can be divided into 3 different categories. The first category is your hormones which actually promote the growth. That is, the first one are called as your growth promoters. They are called as what? Growth promoters. The second one is called as your growth inhibitors. And the third one is called as what? The third one has dual function. That is, they act as both promoter as well as inhibitor. It has both the functions. Here. That is the third one. Now students, if you know, under the promoters we have in total of 3 hormones. We have. The first one is your auxin. The second one is your gibberellins. And the third one is your cytokinins. Under your promoters, we have 3 hormones such as auxins, gibberellins, and cytokinins. And under your growth inhibitors, we have 1 hormone called as your abscisic acid, also called as your ABA, also called as your ABA, abscisic acid, and under both the hormone which has both the functions, that is, it can do growth promoter as well as your growth inhibitor, is called as your ethylene. Is nothing but your ethylene. Is your ethylene, which is also your gaseous hormone, which is also your gaseous hormone. Clear? Now can we go one by one? Can we go one by one? That is, first we'll go with auxin, then we'll understand gibberellins, then cytokinins, then we'll understand ethylene, and finally we'll do ABA, that is abscisic acid, one by one. I'll make sure you write down this information, because I'll be telling you not just the functions, I'll be telling you bioassay, discovery, chemical nature, precursor, every single information from your NCERT. I'll be making you write in a proper format. So write down first one. Friends, write down first for the auxin. So auxin is a Greek word. Auxin word. This particular auxin word is derived. Listen to me very carefully now. I want your 100% attention now. This particular auxin is derived from a Greek word called as your auxin. That is AUXEIN. It is derived from your Greek word. This particular auxin has a chemical nature. Write down. What is the chemical nature? Students, all these chemical natures, precursors are a direct PYQ. Without even thinking, direct question will come from here. Direct question will come from here. The chemical nature is your indole compounds. These are indole compounds. That is the chemical nature here. Now, what is the precursor? The precursor for your auxin. The precursor for auxin is a amino acid. Can anyone in the chat tell me which is the amino acid precursor for formation of your auxin? Precursor. The precursor here is none other than your tryptophan, which is a amino acid. Tryptophan, the amino acid, is the precursor for your auxin. Other than that, is there any element requirement? Yes, sir. The other elemental requirement in the formation of your auxin is your zinc. Is your zinc. Clear? So students, remember, your auxin is derived from a Greek word called as your auxin. That is a Greek word. The chemical nature of your auxin is your indole compounds. The precursor, what is the meaning of precursor? The meaning of precursor is to form the auxin. What is the requirement? The requirement to form the auxin is your tryptophan. Tryptophan is an amino acid. With the help of tryptophan, we obtain the auxins. Now we have auxins. Generally, we have two types. We have the natural auxin, that is indole 3-acetic acid, IAA, indole 3-acetic acid. Then we have indole butyric acid, IBA. Then we also have 4-chloroiodine. This is not in your syllabus, so remember these two. That is IAN, IBA. That is a natural form of your auxin. Now what about synthetic nature? Yes, sir, there is synthetic nature, such as your 2,4-D. This is 2,4-D. Remember, in your biomagnification, in your class 10th, the VD side which you have been learning is nothing but your 2,4-D, that is 2,4-dichlorophenoliacetic acid. Similarly, we have 2,4 or 5 trichlorophenoxyacetic acid. Other than these two, we also have naphthalenic acetic acid. So students, remember, they can ask you a direct question for the natural auxin as well as synthetic auxin. Clear? These are two types of auxins which you will find. So the next part I want all of you to understand is the discovery. Who discovered auxin? Discovery. The scientist, which all of you know. The scientist who discovered the auxin is none other than your Charles Darwin and his son. The Charles Darwin and his son called as Francis Darwin. Charles Darwin and Francis Francis Darwin. Charles Darwin and Francis Darwin are the two scientists who the first time ever they discovered the auxin. Now students, write down these points are very important. The plant which they worked on, the plant which the Darwin and Francis Darwin worked on was your none other than your canary grass. Canary grass. Now this particular canary grass, is it a monocot or a dicot? Is it a monocot or is it a dicot? This particular is a monocot here. Now what is the topic they were working on? The topic which they were working on is your photoperiodism. They were working on your phototropism. Phototropism. They were working on your phototropism. Now what is phototropism? Phototropism is a concept where your particular plant responds to a stimuli. The stimuli which here is nothing but your light. When a plant responds to a light and it grows towards the light, is called as your phototropism. Now what do they do? Let me write down and draw with me now. For example, first initially they took a coleoptile here, a particular coleoptile of your canary grass. They added a light from one direction. The light was illuminated. Yes, there was a unidirectional light. Now when the unidirectional light was from this side, this particular coleoptile started bending towards the light. Yes, it started bending towards the light. Why? I'll tell you later. That is accumulation of auxin in the opposite side. Accumulation of auxin in the opposite side. That's why this particular part is growing more. Hence the bending towards the light. Now, the father and son duo understood one thing. There is something in the tip of your canary grass, of the coleoptile, that it is making it bend towards the light. So what do they do? In the next experiment, they completely cut the tip. That is, they took a plant, they cut the tip. They completely cut the tip. Now the light was illuminated. Now this time the particular coleoptile did not bend towards the light. So they got to know. What they get to know? They get to know that there is something in the tip here which is actually making the coleoptile bend towards the light. Yes, bend towards the light. So in the next experiment, what do they do? In the next experiment, instead of cutting it completely, they covered the tip with a black color cloth. They completely covered it with a black color wrapper. And again, they added a light. Again, they added the unidirectional light. Does it show anything? No. Again, even in this case, whenever there is a black covering on the tip, they got to know that the coleoptile is not bending towards any side. So the first time ever, they discovered that there is something, they don't know what it is, there is something that is making the coleoptile bend towards the light. First time. In both the cases, there was no curvature. If you look at this particular sample B and sample C, there was no curvature. There was no bending towards the light. So they discovered something, that there is something that is making it bend towards the light. But how does it happen? I told you here how it happens. Whenever there is light coming here, the auxin starts to accumulate on the opposite side. Auxin will accumulate on the opposite side of the light. And all of you know, auxin promotes elongation. So the elongation happens this side, and hence this part is growing more. This part there is no elongation. Hence the curvature is happening. Now this was your discovery by Charles Darwin and Francis. Plant, canary grass. What topic they studied? Phototropism they studied. Now, but who isolated? Now, but who isolated the particular experiment? What is something? Yes, they discovered something, but they never told what is something there. The something was actually isolated by your next scientist, that is your isolation of your auxin. That something there was auxin actually. The isolation was actually done by your F.W. Went. F.W. Went was a scientist who did the isolation. Who was the discovery? The discovery was done by your Charles Darwin and Francis Darwin. But the isolation of your particular auxin was done by your F.W. Went. He took a plant. Did he take again the canary grass? No. The plant which your F.W. Went worked on was oats. The same oats which all the bodybuilders eat who goes to gym. The oats. The scientific name of oats is your Avena sativa. Avena sativa, the smallest, sorry. Avena sativa. Now he worked on Avena sativa. Now what did he do? F.W. Went did a simple thing. He also knew that in this growing tip, there is something called as auxin. There is something. But how does he isolate it? So what did he do? Remember, listen to me very carefully. He actually completely cut this. He completely cut open this, and he took this particular tip. Which has all the hormones, that is auxin, and he dipped it. He placed it inside a agar medium. He placed it into a agar medium. He placed it into a agar medium. Now what happens? All the hormone which is present in this tip completely diffuses to the agar. Yes, it completely, the hormone which is present in your tip, the hormone which is present there completely diffused out. It completely diffused out to the agar medium. Now what did he do? He did a simple. He took a coleoptile. This particular coleoptile has no head. It does not have any head. What did he do? He took this agar block and he placed it here. He took the agar block and he placed it here. Similarly, he took another plant. But in this case, he placed the agar block this side. Now listen to me very carefully, students. Listen to me very carefully. If this particular agar block has the auxin here. Now this part will grow more. Yes, this part will grow more because that hormone there called as auxin is promoting the growth. So this particular plant was seen to grow like this. Started growing this side because auxin accumulation is this side, because the auxin is accumulating here. So this is growing this side. Whenever there was a unidirectional light, it started growing this side, opposite side of the auxin. Similarly, this particular block, if the auxin is this side, more elongation happens. Yes. So hence this particular coleoptile, this particular oats started to bend the other side. Because this is the accumulation spot. And hence the curvature happened the opposite side. Now this particular experiment is also the bioassay here. This particular is also called as your bioassay. What is the meaning of bioassay? Bioassay is nothing but a test. Bioassay is nothing but a test to determine at what concentration of hormone does the plant show response. At what concentration of hormone does the plant show response? And hence this bioassay is known as your Avena curvature test. Avena curvature test, which was actually found by your F.W. Went. It was isolated by F.W. went. And this is how they isolated the auxin. But remember students, the first time isolation was also done in the case of your human urine. From the human urine, first isolation was done. Okay, clear? This is the entire test for auxin. So students, let me quickly summarize this. In the case of your auxin, it is derived from a Greek word called as auxin. The chemical nature is indole compounds. Yes, it is indole compounds. The precursor is tryptophan. I hope you have written down. Then we have natural auxins. We have synthetic auxins. Natural auxins like your IAA, IBA, and synthetic auxins such as your NAA, 2,4-D, 2,4,5-T, that is trichlorophenoxyacetic acid. Then the discovery of auxin was done by your Charles Darwin and Francis Darwin. The discovery was done on your canary grass. The topic they were studying was phototropism. What they do? They took a canary grass coleoptile. When they added a light, it was shown that the tip, your coleoptile, was bending towards the light. Yes, it was bent towards the light. Now what they do? In the second case, they completely cut open the tip. Did they see any curvature? No, they did not see any curvature. But in the case of your again black wrapper, again no curvature. So they understood that there is something that which is making the curvature bend towards the light. But the isolation was done by your FW Wendt, that is bioassay, that is Avena curvature test. The plant which was used was your oats plant, which is Avena sativa. Here they cut open the tip. They took the tip and they added into agar plate. Yes, they added into a agar plate. From in the agar plate, all the hormones diffused. Then they placed the block here on the coleoptile. The block started moving towards the light. That is, if the block was this side, the bending was the opposite side. If the block was this side, it bent towards the right. See, if this is the auxin, the growth is happening more this side. So its bending is on the other side. This was the bioassay test, and also the isolation of your auxin. Now, what are the developmental effects? If the auxin is produced, what are different factors which you can see? So first one is your root initiation. Yes, the most important point here is that it leads to root initiation. Then we have cell enlargement. Then along with cytokinins, it also does cell division. It has a feminizing effect. What is the meaning of feminizing effect? It means it develops female carpels. It develops carpels. Carpels is females. It develops carpels. Then it also does flower initiation, which can be used as a commercial application also. Then we have parthenocarpy, the seedless fruits. Then we have prevention of abscission. It is stopping the abscission. What is the meaning of abscission? Abscission means your leaf fall. Like the leaf starts to fall, right? It will prevent the leaf from falling. Okay, it will prevent the leaf from falling. Then the most important one is apical dominance. It shows apical dominance. Now, what is the meaning of apical dominance? If this is your stem, we know apical meristem is here. Then we have the lateral meristem here. Yes, lateral meristem here. This auxin will make sure this lateral growth does not happen at all. Only and only the apical growth will happen. Only and only the apical growth will happen. Have you noticed this? A tree which looks like Ashoka tree or looks like a mango tree which is growing very tall, right? A mango tree which looks like a mango tree but it is not mango tree. It's called as scientific name Polyalthia longifoliales. Polyalthia longifoliales. It is a tree which grows extremely tall. The side branching is actually cut. We cut the side branching. That is called as apical dominance is seen. We add auxin to it. So that is the main function. Now, what is the other commercial applications? I have already told you the 2,4-D, 2,4,D. This particular is a weedicide. What is the meaning of weedicide? It can kill weeds. can completely destroy the weeds. So if the light is in the side of the agar block, what will happen? See that even if the light is there, the complete tip has been cut, right? The complete tip is cut. So the oxygen production is not happening. So usually in this case, not only unilateral light. In this case, light was actually applied from all the places. And then they noticed, since there is no tip, the only hormone which is available there is coming from the agar block. That is why it is bending towards the opposite side of it. Okay? We decide done. Then other commercial application, all of you know, this particular auxin is used in your tissue culture also. What is the application? In the tissue culture, we use auxin for root initiation. That is the commercial application. There is other war application also. How many of you know what is Agent Orange? As how many of you have ever heard what is Agent Orange? Now this Agent Orange was used in a war. Your America and Vietnam War. What is Agent Orange is? Agent Orange is basically a mix of your auxin, type of your synthetic auxin, which will completely clear all the weeds. Whenever there is war, this particular auxin was sprayed from top. If the troop is actually hiding in the forest, you cannot see where to bomb, right? But this Agent Orange, when they sprayed Agent Orange, it completely killed all the dicots. Not mature monocots. The answer to that question I want that in the chat today. That is a very tricky question. If you do not know, next class when I am starting the video, I'll tell you the answer to this question. Next time when I am starting the answer, I'll tell you the answer. That is why 2,4-D mainly acts on your dicots, not on your mature monocots. Let me know in the chat. Okay, this particular Agent Orange completely killed all the dicots that was used in the Vietnam War by Americans, where completely forest was destroyed. They can see all the troops, where are the troops, and they completely bombed them. The other one is your xylem initiation, is not in here. The other ones which are also in your NCERT is your xylem differentiation. Yes, xylem differentiation. It also prevents abscission and also delays senescence. Remember that. It also delays completely the senescence. Yes, it was used in wars. Bioassay done. Applications done. Precursor done. Chemical nature done. Discovery done. So all the points are done for your auxin. Hopefully you can solve all the questions regarding to auxin now. Okay. The next one is your gibberellins. Very important. Now let's study about the discovery first. Let's start discovery. Can we start with gibberellins? Everyone? Any doubt in auxin, you tell me. Any doubt in auxin, I'll repeat again. If there is no doubt in auxin, we will start with gibberellins. Okay. The first point I want all of you to remember is the chemical nature again. The chemical nature, there was indol. Compounds. But in the case of your gibberellins, the chemical nature is terpenes actually. The chemical nature is terpenes. Terpenes or terpenoids. Okay, terpenes or terpenoids. Now, what is the precursor here? What is the precursor? The precursor here is nothing but your acetyl-CoA. The precursor here is acetyl-CoA. Remember acetyl-CoA from your respiration. Acetyl-CoA is the precursor for your gibberellins. Now let's understand discovery. Let's understand discovery. Now this discovery was done by a farmer actually. Farmer and scientist. His name was Kurosawa. His name was Kurosawa. Very funny name, but yeah, Kurosawa. Kurosawa. Now how did he observe this? Listen to the story very carefully. He noticed in the farm one particular maize plant or a group of maize plants was growing very tall. I repeat again, group of your maize plants were growing very, very tall, and they were so tall that it did not have any seeds at all. There were no seeds at all. Now what is the use? What is the use of a plant which is growing very tall but there are no seeds at all? He called this, or entirely we call this, Bakane disease. The meaning of Baka. Baka means in Japanese fool. So this is also called as foolish disease. So he noticed what? This was not in maize, it was seen in rice. Not maize, it's seen in rice. He noticed in the case of rice that it was growing very tall, but there was no rice grains at all. There was no rice grains at all, and hence he called so foolish. What is the point of having a completely tall plant with no grains at all? So he called it Bakane disease, also called as foolish disease. Bakane disease or foolish disease. Foolish disease. Now, later on, after some studies, we got to know that this Bakane disease was actually caused by a fungus. This Bakane disease was actually caused by a fungus, and the fungus name is your Gibberella fujicori. Gibberella fujicori is the fungus name. Can you see this fungus growing at the nodal regions? This is your rice plant. On the nodal regions, this fungus was completely growing. This fungus was growing, and this fungus called as your Gibberella fujicori was producing gibberellins. Because it was producing gibberellins, which was making the plant, rice plant, extremely tall. Extremely tall. This was a fungus. Now, who did the isolation? This was the discovery. Discovery was by your Kurosawa. Who did the isolation? Can anyone chat tell me who did the isolation? I'll write here. The isolation was done by your Yabuta. Yabuta, one more hand, Smuka. What is this? Sumuki. Yes, Sumuki. Yabuta and Sumuki. These are the two scientists who actually isolated the gibberellins. Students, if you want to know how did I isolate, let me know in the chat. Do you want to know how did I isolate? Or do you want to know the bioassay? I'll write on the bioassay, let me know in the chat if you want the how they isolated also. The bioassay which they used was, let me write here, bioassay. The bioassay was very simple. This bioassay called as alpha amylase barley endosperm test. Alpha amylase barley endosperm test. Barley endosperm test. Students, they will not ask you the entire bioassay, but if you want, I'll teach you bioassay. Just say, yes sir, I want to learn the bioassay in the chat, and like the video, I will teach you the bioassay also. Do you want to know? Do you want to know the bioassay for this? Yes, this was out of NCERT. This is not mentioned in NCERT. That's why, see, I want to keep everything within NCERT. That's why, if you know the bioassay, it's more than enough. Students, if you know the bioassay, it's more than enough. Do not burden yourself with extra information. Okay, do not burden yourself with extra information. If you want to know what is bioassay, I can tell you, but let me skip it for now. If you want to know, I'll tell you. It is not in your NCERT. But listen to me for, it will just take 10 seconds to answer this. This is your rice, this is your grain, Yes, it's the grain. The grain has the endosperm. Now this endosperm is diploid or triploid? Everyone tell me in the chat fast. Is it a triploid or diploid? Fast tell me. Endosperm is diploid or triploid? Tell me fast. Here we have the aileron layer here. Aileron layer. Now this particular endosperm starts to produce, when the germination is happening, from this particular endosperm, gibberellin was isolated. From this particular endosperm, gibberellin was isolated. Gibberellins was isolated. Now this gibberellins, what does it do? This gibberellins, how do we know the gibberellins activated? Because this particular aileron layer will produce your endosperm. Now in this test, what do they do? In this test, they found in this test, they basically identified the gibberellins by introducing an enzyme called as alpha amylase. They introduced an enzyme called as alpha amylase. Now what does alpha amylase do? This particular alpha amylase completely broke down your starch to glucose, and this glucose was used for your seed germination. So basically they identified in case of plants, there is a particular hormone called as gibberellin, which is actually responsible for your seed germination. So they isolated it and they did the test. Whenever they introduced more gibberellins, this gibberellins acted on the endosperm, that is, and on the aileron layer, which produced alpha amylase, which completely broke down the starch to glucose, and which resulted in the seed germination. So that is actually also one of the properties of your gibberellins. One of the properties of your gibberellins is your seed germination. Clear? Is germination. And there are more than 100 gibberellins reported. In total there are more than 100 gibberellins which are isolated, out of which the first isolated is your gibberellin GA3. GA3 is the first isolated form of gibberellins. In total we have GA1, GA2, GA3 and so on. In total there are 100 of them. Gibberellin is also called as your gibberellic acid. The first one is your gibberellin A3. General characteristic is what? All the gibberellins are mostly acidic in nature. Yes, they're acidic in nature. And they're either transported through xylem or phloem, and they're formed in the plastids. These are the general characteristics all of you want to know. But the most question oriented here is gibberellic acid, that is GA3, is the first gibberellin to be discovered. The first gibberellin to be discovered is your GA3. GA3 is the first discovered one. Clear? And then what about the function? The first function, which I already told you before, is seed germination. Yes, it is production of seed germination. Other than that, it stimulates stem growth. It completely stimulates the stem growth. What is the meaning? Remember in the Bakani disease, the stem became completely elongated, right? The stem became completely elongated. So it promotes stem elongation. Then it promotes seed germination, I told you. Bolting in rosette plants. What is the meaning of rosette plants? Rosette plants are like your cauliflower. Cabbage, cauliflower. Rosette. In such plants, the stem is very tiny. Yes, the stem is very inconspicuous, very reduced stem. But if you apply gibberellins, to your rosette plants, which can stimulate the stem elongation, that can induce the stem elongation in the rosette plants, and that is called as bolting. Then again, it delays senescence. Obviously delays senescence. Why? It delays the old age. The meaning of senescence is old age. Old age. It delays senescence because I told you already, your auxin, cytokinins and gibberellins, all three of them are your growth promoters. They promote the growth. They do not inhibit the growth. They do not stop the growth. Clear? The next one here is the next one is your other functions, the commercial applications. The commercial application is your, in your NCERT, this is a direct line from your NCERT. The commercial application is increasing the length of the grape stalk. We know the grapes are found in the bunch like this, right? Yes. When you apply gibberellins to it, when you apply gibberellins to your grape stalk, the grape stalk actually increases. It becomes this big. It actually becomes this big. If the grape stalk increases, the fruit will also increase. Yes, that is the commercial application. That is the commercial application of your gibberellins. The increase in length, I already told you, length stem. If the length of the stem increases, obviously more yield. For example, in the case of sugarcane, the entire stem we eat because we have sugar inside it. Yes, if the stem becomes more elongated, means more yield. Yes, it means more yield. So more yield. Elongated, improved shape of the apple. Whenever you applied gibberellins to the apple, the shape as well as the complete improved quality will happen. The shape will, the elongated apple will get. Then speeding up the malting process. What is the meaning of malting process? Malting process is used in your brewing industry to make your alcohols. It can speed up the malting process. Spraying juvenile conifers with gibberellic acid hastens the maturity period. In the case of your conifers, if you apply gibberellins, the entire process, the developmental process becomes faster. The faster developmental process happens. Clear? Is this point clear? Students, this entire thing is given in your NCERT. It is given in my notes. All you need to do is, please make your own notes out of this. The first part of the chapter, I made sure you write everything. From here onwards, the entire PDF will be given to you. All you need to do is write down this, remember the points. Okay? I'll tell you what are the important points. That important points you need to highlight more and more. Okay? Clear? One point I almost forgot here is, in your auxins, I told you it initiates, it prevents abscission. I told you it prevents abscission. Now there's one NCERT line I want to mention here, I almost forgot. This preventing abscission is actually in the case of your young leaves only. In the case of your young leaves or young plants. This is only in the case of your young plants. This abscission I forgot to mention here. This abscission right here, preventing abscission or preventing senescence. Preventing senescence only in the case of your young plants. Only in the case of your young plants. But in the old plants, in the case of your old plants, the senescence is actually increased. What is the auxin doing it? Young plants it is saving. Old plants it is pushing aside. That is the role of auxin, that is preventing abscission as well as preventing the senescence. Senescence is prevented in the young plants, but in the old plants, it is not prevented. They are pushed out. Old leaves are completely pushed out. Okay, that is the point here. I forgot to mention. If your NCERT, I'm trying to remember your NCERT. Did I miss out any point? The other point I think I missed out in your auxin is this auxin actually is involved in your hedge making. Do you know what is hedge making? If you watch some cartoons, or if you go to tea gardens, or if you go to Sri Lanka, if you go to your Sikkim, if you go to your Darjeeling, they make tea gardens, right? It's so uniform. What do? They completely cut it, right? They completely cut. That is because of your auxin. It makes sure that only apical parts develop. Only apical parts develop. Gibberellins is, I think, is over. Then is cytokinins. Is gibberellins clear to all of you? Is gibberellins clear? Let me revise one more time here to all of you. Gibberellins, the chemical nature is terpenes or terpenoids. In gibberellins, the chemical nature is terpenes or terpenoids. Precursor is your acetyl-CoA. Then we have discovery was done by your Curacao, where in the case of your rice plant, that is Bacillin disease or foolish disease. What was infecting? The infecting agent was your fungus, that is your Gibberella fugicori. Then bioassay is called as what? Bioassay is called as your alpha amylase barley endosperm test. Then what are the types of gibberellins? In total, there are hundreds of gibberellins. That is, more than 100 gibberellins have been isolated. We call them gibberellin A, gibberellin B, gibberellin C. No, gibberellin 1, gibberellin 2, gibberellin 3. Out of all the gibberellins, the first discovered gibberellin was your gibberellin A3. Sorry, gibberellin GA3. And remember, all the gibberellins are acidic in nature. Then they are transported either through xylem or phloem, and they are formed in your plastids. They are formed in your plastids. Then what are the functions? Stem elongation. Yes, Bacillin disease, you saw the stem was completely elongating. Other than that, we have promotion seed germination, bolting of rosette plants. The rosette plants completely bolt. Then we have your delayed senescence, that is old ages completely delayed. Then it increases the stalk of your grapes. Yes, grape stalk elongation. Then we have increased length of the stem. In the case of your sugarcane, saccharum, the stem elongates, more yield. Then we have elongated and improved shape of the apples. Then speeding up the malting process, and spraying on their conifers will hasten the reaction of your juvenile phase to your mature phase. Clear? Any doubt here? Any doubt here, students? Once again, let me see the chat. Any doubt here, you can ask me right now. Can we go to cytokinins? Can we go to cytokinins now? Can we go to cytokinins? Now let me tell you similarly, I'll tell you what is the chemical nature. Write on with me. The chemical nature of your cytokinins. The chemical nature of cytokinins is your purines. Purines, such as your adenine. Adenine is a purine. Yes, adenine is a purine. So it is a adenine derivative. It is a chemical nature is mostly purine in nature. They are mostly like your adenines. They are mostly like your adenines. Then what about your precursor here? Precursor here. In the case of your precursor is nothing but is your tRNA. Transfer RNA is your precursor here. Now who discovered this? Discovery. The discovery was done by your Skoog. Discovery was done by your Skoog and co-workers. Skoog and his co-workers. Skoog and its co-workers. Now this is your Skoog. Yes, Skoog was the first person who discovered the cytokinins. What did he do is, Skoog was working on your tobacco callus. Skoog was working on the tobacco callus. So he was applying auxin to the development of the callus. And all of us know auxin will promote your root elongation, but cytokinins will promote shoot elongation. Very important point. Then what did he do? He observed that tobacco callus proliferated. What is the meaning of proliferation? Proliferation means the tobacco callus became very big. It is able to grow rapidly. When we added along with auxin extract of your vascular tissue, yeast extract, or coconut milk, or herring sperm DNA. When we added any of the following along with auxin, the complete growth of callus happened. What we understood from here? He understood there is something in all of these different substances that is called as your cytokinin. That was your cytokinin. So he first discovered it, but isolation was not done by this guy. Isolation was done by your Miller. Isolation was done by your Miller. Isolation was done by Miller et al. Isolation was done by your Miller. Discovery was done by your F. Skoog. Discovery of Skoog, but isolation was done by your Miller. So how did he isolate? The Miller, can you see this friendly looking guy? Then he showed that DNA had powerful cell division promoting effect. Miller et al. Later identified and crystallized the cytokinins promoting activity substance that is termed as kinetin. He completely isolated from a herringfish sperm. From the herringfish sperm, he isolated kinetin. But that was in the case of animals, not in animals. That was the case of fish. But what about plants? He isolated even from plants. He isolated from your maize. In the case of your maize, in the case of Zea mays, he isolated a substance called as zeatin. He isolated zeatin, which is almost similar to kinetin, which is a type of cytokinin. So students, remember the discovery was done by Skoog, but the isolation was done by your C.O. Miller. Where in the case of your herringfish sperm, there was the isolation, kinetin, but in the case of plants, it was done by your Zea mays. What am I writing here? In the case of Zea mays, the isolation was done and we called it zeatin. Then, what are the functions? What are the functions of your cytokinins? Now, I already told you first function is your tissue culture. The first function is your tissue culture. Remember, your auxin will promote your root elongation. And if you add a lot of cytokinins, cytokinins will promote shoot elongation. What if you add in equal quantity? What if you add equal amount of your auxin and cytokinin? Then we have a proper callus development. See here, cytokinins does cell division. That is the main function. Cytokinins along with auxin promotes cell division. Natural substance with zeatin. What are the functions? Regulates cell division. That is the first point. Along with auxin, it promotes rapid cell division. Modifies apical dormancy and promotes lateral growth. Did you see that? Your auxin is promoting apical, but your cytokinins is promoting lateral growth. Complete lateral growth promotion is done by your cytokinins. It delays senescence. Yes, it is a growth promoter, hence it will delay senescence. Promotes nutrient mobilization. From here to there, mobilization nutrients. Promotes chloroplast development. Yes, it helps in development of a chloroplast in your plants. Helps to produce a new leaves and adventitious shoots. Also it helps in. So those are the different applications of or roles of your cytokinins. Hello Gopika. Gopika ma'am is here. I see. Now what about ethylene? The last two is your ethylene and abscisic acid, and then we are done. So ethylene, students, the first point all of you should know is ethylene is your gaseous hormone. That is the most important point. The ethylene is your gaseous hormone. Now what about your precursor here? The precursor is also an amino acid. Remember, in the case of your auxin, it was tryptophan. A question can come like this: which are the two hormones where the precursor is an amino acid? The answer is your auxin as well as ethylene. In the case of ethylene, the precursor is your methionine. Methionine is a precursor here. That is an amino acid. Now what about discovery? The discovery here was done by your H cousins. H cousins were the discovery here. How did they discover? They took an orange. They took a particular orange, and this particular orange was completely ripe. This orange was completely ripe, complete ripe orange. Now this particular orange was kept along with green raw banana. Was kept with green raw banana. Now they observed one thing. What they observed? This particular orange was releasing certain gases. Yes, it released certain gases, which converted this particular gas, completely converted this particular raw banana into your ripened banana. So after some time, this banana ripened. It's ripened banana. That was observed by your H cousins. That is the discovery of your ethylene, which is a gaseous hormone. What is the precursor? The precursor here is methionine. Methionine, which is an amino acid, is the precursor. Now what about bioassay? The bioassay here was, the test was here triple response test. Triple response test. That was the bioassay which was done here to understand what amount of concentration of ethylene if plant produces, which will promote the fruit ripening. Clear? Now what about the advantages? What are the different applications of it? Now I have divided application or effects of ethylene on each and every specific part. For example, if you notice the effects of ethylene on seed and seedlings. On seed and seedlings, it will break seed dormancy. It will completely break the seed dormancy. It initiates germination. Yes, it is promoting dual function. Remember, your ethylene has a dual function. Then it horizontal growth of seedlings. Yes, horizontal sort of seedling will happen. Then we have swelling of the embryonal axis. The embryonal axis will swell up. Then we have apical hook formation in the dicot seedling. In the case of your dicot seedling, this is your seedling. Your hook will develop like this. That is your apical hook. This apical hook will start to develop horizontally. This particular hook will develop horizontally, and after some time, this apical hook will become what? Completely expand. This apical hook will also expand. This is actually nothing but your triple response. This is the triple response assay. This is the triple response assay. Yes, it is also used for your tomato ripening. Not only tomato, even in the case of apples, even in the case of papaya. You might have noticed, if you have one papaya which is extremely ripened, amazing smell is coming. If you keep that papaya along with other papayas or other fruit, that fruit will also ripen very quickly. Yes, it will ripen very quickly. That is the gas, that is ethylene, the gaseous hormone which is being released by the papaya. So this is the complete effects on seeds and seedlings. Now what is the effect on the shoot? The effect on the shoot is very clear. It breaks the bud dormancy. Yes, it is promoting the growth. Sprouting of the potato tubers and internode and petiole elongation in deep water rice plants. In some of the plants which are completely submerged, even in aquatic plants and also submerged plants, it was doing shoot elongation. Now till here, it is completely your growth promoters. But students, you need to know this particular also has inhibitor also. Before that, let me see the root. On the root, it promotes the root growth. It also does root hair formation. Also does what? Root hair formation. Remember, this is a difference. In the case of auxin, it was root development. But here it is root hair development. That is the difference which I want all of you to remember. root here, development. Okay. Sprouting meaning, sprouts means, have you noticed? If you take chana or something, and if you keep it in green gram or your brown gram, Bengal gram, you keep it in water. Keep it in water or little bit of water. Next day, from the particular sprout, from particular gram, one yellow color something is coming out, white color something is coming out. That is called as your germination of sprout. That is also called as your sproutling. Okay. Now, what is the effect on flowers? It induces flowering in pineapple and mangoes. Yes, it is flowering. Promotes female flowering in cucumber and hence increases the yield. That is feminizing effect. Then, what about the effects on fruits? All of us know in the fruits, it is commercially used to ripen fruits. That is your epitome, which is a commercial application which does the fruiting of your fruits. That is your green banana to complete ripen banana. That is effects on fruits. Now, see, did you see everywhere I've mentioned what is the effect on your shoot? What is the effect on the seedlings? What is the effect on roots? What is the effect on flowers? What is the effect on your fruit? But what is the effect on your various organs? In general, it does inhibition. What is inhibition? On your leaf, on different branches, in different cells. It does senescence. That is, it causes the old age. It causes the old age. It also causes the abscission. It also causes the abscission. That is, it makes sure the leaf is falling. It makes sure the leaf are falling. So students, remember, in the case of your ethylene, it has a dual function. Your ethylene can do both. That is, promote as well as inhibit. So I hope that point is clear to you. What it happens on seedlings, root, shoot, flower, fruit, and also various organs. Clear? Is it point clear? Can we start with abscisic acid? All of you? Can we start? Opposite to auxin. Yes, it is opposite to auxin. Seed dormancy meaning it is making sure the seed becomes dormant, means to make sure the seed is not germinating. If it is breaking dormancy, it means the seed is germinating. It is keeping the seed dormant, means the seed is not germinating. Seed dormancy is the seed is becoming dormant, it is hibernating, the seed is not germinating. Okay? Amazing. Students, entire notes will be provided to you, all this information. You can write down all of this. All of this is important. Please write down all this. Notes will be given to you. The PDF will be provided to you. Deliberately, if I write every point of your hormones, it will take more than 2 hours. It will go for 1.5 hours. That's why the roles of hormones, I am discussing you the initial part, the effects, which is mostly your remembering thing. I want all of you to write down that. Okay? Now, the last part is your ABA, also called as your abscisic acid. Now students, if you remember, I told you in plant growth, we had 3, which promotes growth: auxin, cytokinins, and gibberellins. Ethylene has both the functions, that is, it is promoting and also inhibiting. But in the case of your abscission, it is the hormone which makes sure the plant will not keep on growing. Yes, if there is nothing to keep the plant in check, the plant will keep on growing, right? This is the checkpoint. This is the hormone which will make sure the plant stops to grow at one point. That this particular plant stop growing, next part is just growing, it will go and stop that also. So this is the evil part. So what is the chemical nature? The chemical nature again. The chemical nature here, one second, let me write down, changing the color. The chemical nature here is carotenoids. The chemical nature in the case of your abscisic acid is carotenoids. Is your carotenoids. Carotenoids. All of us know this carotenoids. Carotenoids. Remember, that's all. It's a extra information not required. carotenoids. Now what is the precursor here? The precursor here is, it's a chemical called as your zeaxanthin. Xanthine. This xanthine is a type of your xanthophyll. Is a type of your xanthophyll. Now this is xanthine is the precursor for formation of your ABA. Now what about discovery? Now there were a couple of scientists who were working. They were working on different chemicals. They found that one particular chemical is inhibiting. They call it inhibitor beta. One particular scientist called it abscission 2. One particular scientist called it dormin. But later after some years, they understood that the chemical nature of your inhibitor beta or abscission 2 or dormin is nothing but the chemical nature of all the three is nothing but your abscisic acid. That's how it was discovered by multiple people. That is called they called it abscisic acid. Now what is the functions? The function is very simple. This particular abscisic acid is also called as your stress hormone. It's called as what? Stress hormone. Now why is it called stress hormone? Listen to me very carefully. Whenever there is stress, what type of stress? Whenever there is no water. Whenever there is no water, this particular hormone called as your ABA, abscisic acid, will go to your stomata and it will tell the stomata, you better close now. There is no water for you, please close your gates. So your abscisic acid will completely close the stomata, which will make sure that less water is lost with in terms of transpiration. So hence it is called as your stress hormone, which actually helps the plant to control the water loss. Increases the tolerance to stress. General plant growth inhibitor, it completely destroys the plant. It is make sure the plant is not overgrowing. Then inhibitor to the plant metabolism. Yes, to make sure the metabolism is little less. It helps in seed development and maturation. Complete opposite to your gibberellins. Remember, gibberellins cause seed dormancy. This is seed germination. Complete opposite. It is antagonist to your gibberellins. It promotes seed dormancy. It is making the seed dormant. But your gibberellins was seed germinating. But this is making your abscisic acid is making seed dormancy. That is, it is inhibiting of germination. Abscisic acid is making sure the seed germination is not happening at all. Germination. It stimulates the closing of stomata. Yes, I told you, whenever there is stress, stomata closes, and it is complete opposite to your gibberellins. That is the functions of your abscisic acid. Any doubt here? Why is it called stress hormone? I told you why is it called stress hormone. I only see 91 likes. I want everyone to like the video right now before I tell you the small table. See here, this is a table which I want all of you to understand. In the case of auxin, it is a plant growth promoter. Root initiation, flower initiation, parthenocarpy, seedless fruit. Then prevention of abscission. Prevention of your abscission. Apical dominance, yes, to make sure one is dominant. Then its feminizing effect, cell division along with the cytokinins, and cell enlargement. What about gibberellins? Gibberellins stimulates stem growth, promotes seed germination, breaks bud dormancy, delays senescence. Then we have your cytokinins, regulates cell division, then modifies apical dominance and promotes lateral dominance. Then we have it delays senescence again, along with your cytokinins, auxins and gibberellins. They completely promote, that is, They delay senescence and promote the growth, promotes nutrient mobilization, helps to produce new leaves and adventitious shoots. Then ethylene breaks seed dormancy, buds dormancy is broken, sprouting, internode and petiole elongation in deep water and rice plants, promotes root growth, induces flowering, commercially used in your ripening of the fruits, promote senescence and abscission. That is, your ethylene shows dual nature. It shows dual nature. Then we have lastly the abscisic acid, increases the stress. That is, it helps in against the stress, against the loss of water. It promotes seed dormancy, against the gibberellins, and it inhibitor of plant metabolism. Those are the different roles of all the hormones, the 5 hormones which is given in your NCERT. No more questions will come out of it. I have told you bioassay, I have told you precursor, chemical nature and the discovery. Every single hormone we have done. That's all will be asked in your examination. Nothing more than that. Trust me, will come in your NEET 2024. Students, before I go, let me tell you about a Telegram channel, that is your Vedantu NEET Telegram channel. On the Telegram channel, we do weekly schedule, daily quizzes, weekly live session, daily session PDF, weekly mock test, and Super 6 series will be given to you. This is our screenshots of a Telegram channel. On this Telegram channel, we are providing all the notes, doubt clearing, everything. You can also join my Telegram channel. That is, if you just type my name, that is Basavaraj sir, you'll get my name. Just type Basavaraj sir, you'll get my name. And how do we do the PYQ quiz? Now students, now you finish the chapter. Now you have finished the complete chapter, done and dusted. Yes, I want all of you to comment today. All of you to comment in today's video. Please comment, go on. Just type plant physiology, done and dusted. All of you go on today's video. Just write plant physiology, done and dusted. If you go to your PYQ quiz, go to description today. In today's video, if you go to description, there is your link. Click on the link. You get a link and you click on the link and start the test. That is last 7 year PYQ, completely free from this chapter, is given in the description. Go on. On the description, complete 7 years, last 7 years, and you will get a performance report also and solution. Not only questions, last 7 year PYQ with solution will be given to you on the description here. Okay, and in Telegram channel, you can use a bot here. We have a bot which can help you to give the Telegram quizzes every single day just for all of you for completely free. Telegram channel is Vedantu NEET English. Link is in the description. Go on, join the Telegram channel. You can scan this code and also join. And in the end, I want all of you to say one thing. Thank you so much, students, for attending today's amazing class. I'll just bring on the screen. I'll show you some of students. Here, if you go to the description here, see here, solve NCERT based Telegram quiz from this chapter is here. Then we have last 7 years quiz for your chapter. Click on this, you'll get it. See here, add your phone number, email ID, you do it, you get the quiz, and you get the quiz, and then I think that went away somewhere. Somewhere. So you can get it to the quiz. Everything is in the description. So thank you, students. Let me know in the comment section how the session was today. And I believe your plant physiology is completely over, completely done. We started off with your photosynthesis, then we did respiration, then we did the plant growth regulators. Next we'll be doing morphology of flowering plants. I know we did plant families before, but I want everyone to finish class 11th first. With your morphology and anatomy, your complete class 11th will be over. After this, we'll be starting your 12th board series, where I'll be teaching you along with board syllabus and complete 12th syllabus. If that is okay, let me know in the comment section how today's class was, if you understood everything, and I'll see you in my next class, that is morphology of flowering plants. Take it along.